Okay, can start now?
Yes, you may start, right?
Assalamu alaikum. My name is Raad. My supervisor, Dr. Noor Shaida Afin. Today, my project, cloning and expression of the Giardia lambelia recombinant protein SH22 in E. coli. This is my autoline. The Giardia lambelia flagella. The Giardia lambelia have the two species, Giardia intestinal, Giardia duodenals. Is in the intestinal pathogen is the major cause of diarrhea. The Giardia lambelia, the three major causes, the third most common agents causes the diarrhea involve this symptoms and diagnosis. The symptoms, the Giardia, chronic diarrhea, abdominal pain, weightless, and the diagnosis, Giardia, microscope stool analysis, antibody test, PCR, polymerase chain reaction. The research aim to produce recombinant protein host light. The grant work futures research aim explore the potential and the application in the diagnostic in giardiasis. A specific objective to produce recombinant protein SH22 in bacterial host. Assess the solubility and purity of the recombinant protein. Detect the antigenicity of the recombinant protein SH22 protein by Western blot. SH2 in auto-prefer study, Western blot analysis identify 21 potential immunogenicity bind the trophozoite protein with the SH22 domain contain a protein bind the interval significate the protein was select recombinant produced in E. coli slide a future study in the potential and role giardiasis. The protein expression in E. coli required suitable host BL21DE3 for the eukaryote protein expression factor, including a promoter, the control protein expression, in this case BET13A. The method and sample collection. Transformation. I used the transformation BL21DE3 BL21DE3 culture the bacterial and overnight with during take the 1 mole for bacteria, make the transformation with the heating factor. Expression. This is expression stay for my methodology. And the purification, use the use the different concentration for the imidazole. Making the purification antigenicity evaluation may use the Western blot state. Result. During the make the transformation, I'm using two type of the bacterial. Use the BL21 and the top 10. Make the transformation in BL21. make the transformation with the antibiotic kanamycin, successful the positive positive and not the growth in positive, not in control growth because find the because the kanamycin. This is the BL top 10 successful, same the BL21 and the negative control not growth. After make the transformation, check my colony by blue-white screening. You can see in the colony found the blue and white. After the make expression, choose a new colony for white, make the expression. This a protein expression. During make the protein expression with the antibiotic, make the optimize of the time. Make the optimize of the time the expression protein. Collect the sample, the first one after the after and before the added IBTG. First 1 hours, 2 hours, collect a sample, make the SDS page, check the my protein time, make the expression. This is my Western blot. After the expression, make the Western blot, check the time for optimize for check the optimize sample collection by Western blot. This is Western blot. Protein purification. Protein purification, I'm using the different concentration for imidazole. 10 imidazole, 20, and this list for the concentration of the imidazole. You can see the purity for my protein in high concentration for imidazole. This is Western blot purification. You can, after the first one, the Western blot, not make the purification. Now the Western blot would make the purification, the purity protein. You can see my protein in Western blot. After this, make the protein concentration. During make the protein concentration using the four concentration for imidazole. Check the protein concentration for 1 imidazole for all the concentration in imidazole. Conclusion: the successful for transformation of the BET plasmid in E. coli confirmed through selective potential. A protein expression evaluate using the SDS page and Western blot technique with the results show a grand expression sample between the S2 and S9. Western blot result related particular and the 250 imidazole concentration was found to be obtained purify SH2 protein with the minimum concentration for other proteins. These find contribute understand the role of the SH2 protein as the protein diagnosis make the Jardia. There was a future research on the diagnosis application. This is my reference, and thank you. Okay, Doctor. Doctor.
Yeah. All right. Thank you very much.
All right. Good.
Yeah.
Thank you, sir. Hello, can you hear me? Good morning.
Good morning, Doctor.
So, thank you for your presentation, and I saw that you have great to achieve your achievement, your objective, right? Because basically you have 3 objectives there, and then you want to test on the indigenicity. Then you finally are able to produce the protein in the vegetable, and then you show that there's a binding with the antibody. And I think that you put a lot of effort and then to get this kind of achievement. So can I know that okay, the first question, can you enlarge your slide again, please?
Last slide.
Yeah. Put them in the slide format again. Go back to the slide. Yeah,
Okay.
you put them in. Put the slide into a presentation mode.
Okay. Any slide, Doctor, for the Western blot?
No. You show your slide in the presentation mode like just now. Because it's too small, I can't see it. You turn it into the presentation mode.
Right, just make it bigger.
Doctor, okay, any slide you need.
Okay, doesn't matter. Okay, can you stop at this slide? Start on this slide. Okay. Can you explain to me what is the difference between the why do you want to use the E. coli 21 and also the top 10 cells?
Okay, because the BL21 using the protein expression, because the BL21 have the 7 promoter. The BL21 have the 3 promoter. This using the reply the plasmid.
Then top 10. Why do you want to use top 10?
Top 10 have the 3 promoter.
3 promoter. Okay, which cells do you use?
Use the cloning. They use the cloning and transformation only.
All right. Okay.
Not the good using and make the expression, but the BL21 have the 7 promoter can take the good result for the make the protein expression.
All right. So your study, can you go to another slide in the SDS page, please?
Okay.
SDS page. Yeah, this one.
Yeah,
this one we can see that you already do your expression, and then from this gel, why do you want to use the different kind of the concentration of the IPG?
Yeah.
Why do you want
because I need to check for the time, make the expression, make the optimize for the time. I need to check the good time, make the expression for my protein.
Okay, since that this is your answer, okay, can you go to the next slide, please?
Okay. Yes.
This one. All right. So we can see that before the purification, right? And
No.
can you see that there is a double band? Maybe start from the. It's too small. I can't see it clearly. It start from an S3, S3, S4, S5 until S7. Can you see there are 2 bands there? And can you explain what is that band?
Yeah, this is for the something you can make the because heating, because not the impurity my protein now, because the quantum. You can see this is after the purification, the protein, my protein impurity. You can see found the 1 band. This is before the purification.
Yeah, I know that this is before purification, and because that I was wondering that during in the negative control, you see the S0 negative control E. coli. There's still a very thin band there, right? Negative control.
Yeah.
And
I think.
Do you know what's that band?
Yeah, I know. I told you. I think because the heating, because the quantum, because this first one make the Western blot. Because this is not with the purification the protein.
Okay, can you go to the purification part, please?
Yeah, this is a purification.
Okay, and we can see that during the purification, and then it seems like all right, hang on. You compare to the positive control,
Yeah,
positive
control.
control, negative control is so nothing, all right. And the 10 millimolar, we still can see. Okay, that's the size seems like a little bit different. And positive control, what kind of the reagent do you use for the post control, and why the band they are not so specific compared to your result? What is the post control do you use?
BR, Victor, BR, I don't know. I don't know now. But BR, the BR.
So, maybe you still can you please go to your this slide and then go into details. You compare your lane at 10 millimolar and the 250 millimolar. You can see the band there, and then why is the band in the 10 millimolar so more intense compared to the 250? Then another thing is it looked like the band at the 10 millimolar and the 250 they didn't show in the same size, because the size of the 250 seems like much smaller compared to the 10 millimolar. Can you explain this?
Yeah, because this is the first one that use the purification, because the last the imidazole you find last amount for the my protein in the nickel resin.
Okay, then why the
See, you can see here.
yeah, we can see here.
Yes.
Yeah.
Yeah. This is the different size in the during the make the protein purification.
Yeah, can you go back to the Western blot just now?
Yeah.
Yeah, this one. Okay, I was thinking that you already got your protein, and how do you verify that this is your protein? Because when you compare to the post control, it seems like there's a lot of the massive band there, and compared to your purified 250 millimolar, and you put an arrow saying that this is your protein. How do you ensure to claim that this is your purified protein?
Victor, you can again.
How are you sure that this is your purified protein?
Yeah, because I'm using ladder. You can check the size for protein in with the control with the 10 imidazole, 20 imidazole.
Well, if the size is correct, and normally the post control is quite important because it can use as a comparison to your specific protein. But right now, your post control, we can see there's a lot of band there. Can you see the post control? Right?
Yeah, I can see.
Yeah. Normally, the post control, they must not, in general, it's supposed to show a same band compared to your 250 purified protein. But right now, your post control show like a series of band, and there's no any specific band compared to your samples. And how do you explain it in your thesis?
Yeah, Doctor, this is because after the make the 10 imidazole and the 250 imidazole, different the concentration for a protein. You can see in SDS for the protein purification. Because the different size for band, because the different amount the protein during make the wash resin by the last concentration for imidazole.
All right. Okay. Can you go to the slide of the references, please? Okay, and I think that you put all right, the sentence is okay. All right, and the conclusion, please. Conclusion.
Yes.
Okay, last sentence. You said you claim that you already you concluded that you got something and then it can be and proceed into the diagnostic platform already. Okay, from your recombinant proteins. From your study, can you conclude that what is the step? What is the further step needed to determine to be identified before turn your protein into a diagnostic application?
Yeah.
Right? Okay.
Yeah, Doctor.
What's the next step needed?
Yeah. Found the other method using the diagnosis for the Giardia, but I need the specific diagnosis for the Giardia. You can use it the MyProtein, make the tools. You can use it check the infection for the Giardia very fast, very fast. Uh, uh, very fast because the other diagnosis need more time, expensive, and something. But this is, you can make rapid test, use the direct diagnosis for the Giardia and the easy and the cheap the application diagnosis.
Okay. All right. Thank you so much, Raat. Okay. Thank you for having a very good explanation. I'm done. Thank you.
Thank you, Doctor. Thank you.
I'm done. So who will be the next speaker? Yeah,
Okay.
Stana, right? So thank you so much to Stan and also to your sponsor, Dr. Sida. And we hope that to see you soon in your next presentation for during your Viva. Okay, good luck, Stan. Good luck.
Okay. Good luck, Doctor. Thank you, Doctor. Thank you.
All right, thank you. So the next presentation, are you ready?
Only one evaluator.
Yeah, I guess how many are evaluated? Let me check. Or maybe if there's anyone want to ask, Raat also can raise your question here. If any lecturer or anyone who would like to have any questions. Let me check. One only, and also Cairo, sorry, Dr. Cairo also the examiner, Dr. Cairo. Raat.
Okay, Doctor. Okay.
Yeah, Dr. Cairo, please.
Oh, thank you, Hern. Thank you, Dr. Hern. Right, thank you very much for presentations. Okay.
Okay, Doctor. Thank you.
Yeah, actually, I have a few questions, but it's already asked by Dr. Heng.
Okay.
Perhaps just want to add some more questions. For your expression, why you
Yeah.
choose only 6 hours interval periods of expression? Why don't you go for longer, like 24 hours?
Doctor, because this is my protocol, I think in 6 hours. I discussed for this with my supervisor. Make the 6 hours.
6 hours. Do you think that 6 hours enough for after you do the experiments and you did the Western blot? Do you think 6 hours is enough for you to express the protein?
I think enough because you can see my protein in Western blot in this time.
Because yeah, in order to expression is one of the experiments. Another one is you want to quantitate the proteins, right, and you want to use it for your diagnostic. So that's 6 hours enough for the protein to be used for your diagnostic purposes. If you look at your slide number 14, this Western blot, okay. So I just wonder at 250 millimolar if imidazoles, is this you purify the pellets or supernatant?
Supernet.
Supernatants. Is this
Yeah.
based on 6 hours or what? What
5 hours.
5 hours? Why you choose 5
Yeah.
hours instead of 6 hours?
Because I'm during the expression for my protein, the successful in 4 hours. I'm making the 6 hours. You can check. You can use it the 5 hours because already my protein make the expression during the 5 hours, between the 5 hours and 6 hours. You can use it between him in 5 hours.
Sorry, Dr. Khairul. Ra'ad, can you show the slide for Western blot, slide 12?
Okay, this doctor.
Okay, yeah. And then explain now why you do 4 hours, 5 hours instead of.
Yeah. Dr. Herol, see in C4 and C8, you can see the my protein expression during make the Western blot.
Yeah, I can
The
see that.
good expression. You can choose this time because already in 5 hours and in 4 hours already my protein expression.
Yeah. Because you said that you are for your another purification, you choose 5 hours, the S7, right? But at your S8, 6 hours is also produced. If you say that your protein already produced, actually at 3 hours or 2 hours is already produced.
Yeah, my protein produced in 4 hours and 5 and 6 hours.
Look at
You can
your Western blot, 12, slide 12.
see in 3 and 2 you can see is band into is very small in 3 and 4 and 5.
S4. Okay, so if you look at your S4,
Yeah,
okay,
as for
so which one the product that you want? Is that the upper one or the below one?
the other one is first one.
Yeah, which one? The upper band or the below band?
The first band. I
The
don't
first
know.
ones, right?
Yeah.
So if you say that you are choosing 5 hours, which is S7, actually at S4 is 2 hours, it's already expressed.
Yeah.
Yeah,
I'm using the range between.
okay, doesn't matter. Okay.
Mhm.
So because for your purification, it seems that at S7, at 5 hours, is very thin band. So I'm thinking, I have some thought that perhaps is it possible that your protein has no optimal condition to be expressed during 5 hours, or perhaps the condition itself is not conducive for them to express the protein. That's why you got very low protein instead of a bulk protein. Any reasons perhaps you want to add? Any reason why you get very faint protein for 5 hours?
Doctor, you can see in Western blot in 5 hours is that my protein in find in 3 hours find in 6 hours find.
It's
I'm
very
I'm
faint.
choosing the
It's very faint. It's
Uh,
very faint.
Yeah, may I interrupt, Dr. Khairul? Okay, now
Uh-uh.
you cannot be so defensive when people ask you comment on your work. It's clear that on 4 hours the band is a bit faint rather than 5 and 6 hours. So it's actually Dr. Khairul. There's no reason behind this because Ra'ad he just did a very few work and then he's now in Iraq. So I think this now shows that right, you need to do more lab work in improve your result. Okay, so
Yeah.
obviously you need to be here and you need to return here and do your lab work.
Yeah, because you have to show us the expression is really works. So if you produce this faint band, it seems like the expression condition is not conducive enough for you to express the protein. So if you can produce a bulk proteins, I think it's much better. So I think you need to extend the time hours for the expression. Can we go for next question is on your preparations of the BL21 for the transformation process. So how you prepare your competent cells of BL21? So what is the method that you use for competent cells preparation?
You need the method?
What that you use to prepare to make sure that your cells is conducive enough to receive the foreign or vectors. What's method?
Doctor, you mean method for transformation,
No,
the BL?
I'm asking you what is the preparation for the BL21 you did to make sure that the transformation is work.
The BL21 using
the competent cell preparation. How you do the
okay, how do you do the expression for BL21?
No, competent cells. You know what is competent cell?
Competent cell for the make the transformation, the making cell.
Yes. So how you do the competent cells?
I'm already my supervisor take me the BL21.
So
Make the
how you make sure that the BL21 has a very permeable membrane in order to receive the vector or plasmid.
I'm culture the BL21 overnight. Use the 1 mol for the BL21. Make wash by CL and heating the in transfer my plasmid incubator 1 hour.
Okay, thank you. I got the answer. So you are using calcium chloride methods, for your transformation or your competent cell preparations.
Yeah.
Okay. So on Dr. Heng's question just now, you mentioned about this slides that you choose top 10 and also BL21. So you said that on top 10 there are promoters and BL21 has a promoters. So may I know what promoter that you mentioned on the host itself?
And BL21 have the 7 promoter.
What promoter that you mentioned? What promoter that you mean? What promoter?
The promoter for the RNA, DNA gene.
Can you explain more?
The doctor, the BL21 using make the expression because they have the ability successful for the expression because the BL21 have the 7 promoter. The other types
what promoter that you mentioned?
RNA and DNA.
Yeah, what is the name of the promoter?
7 promoter RNA.
Okay, I think sorry, Dr. Khairul, I just want to add out a little bit to you. And what Dr. Khairul asking the name of the promoter. You keep mentioning is a 7 promoter. What is the name of that promoter?
Poly promoter.
The name of that promoter. Do you know that?
I don't know.
All right. Maybe you need to find out and
okay,
your thesis. Thank you, Dr. Khairul. Thank you.
I think one more. for your Western blot,
Yeah.
any Western blots, okay? So what is your primary antibody? What is your primary antibody?
I'm using the primary antibody for the histag.
Histag, okay.
Yeah.
So for the secondary antibody,
Wait, I don't- um, I'm already connect with the histag, the secondary and primary.
okay. So perhaps last. So for your blue and white screening, so if you can look at your blue and white screening, okay, it seems like your blue and white screening, the populations of your bacteria is so condensed. So how actually you choose your positive bacteria, positive single population?
I'm choose white bacteria, good colony. I'm not choose the blue bacteria.
So are you sure that your plate has no contamination? Because if
Yeah.
I look at your plate,
Because
there are
Mhm.
two populations there. There are big colonies and also single colony, very tiny colonies. Any reason that you have multiple size colonies?
No, I'm using this not contam. I'm using the same my colonies. I don't know why this where the difference between the size the colonies.
So how you spread your colonies? How you spread the culture?
I use I'm choose the active colony in the during the transformation. After this, I'm already making the plate with the antibiotic kanamycin, making the 2.0 microliter by the XGAL, making the separate in the plate. After this, make the separate for my colony in the plate.
Okay.
Wait overnight. This is my result.
But I think if you want to use this picture again for your Viva, you have to come up with explanation why you got lots of multiple colony size instead of
Okay.
synchronized colony size, because I'm afraid that there are contamination. Okay, that's all from me. Thank you very much, Ra'ad. Okay, Dr. Heng.
All right, thank you so much, Dr. Khairul, for your very good question. And I think Raad kindly accept the good input from Dr. Khairul and then to improve your thesis. And I think that your supervisor, Dr. Shida, is a very well experienced researcher in this kind of the study, especially for the diagnostic prediction of the COVID-19. So try to improve your limitation in your work. As your supervisor mentioned that you are not away, and some of the experiment, it seems like you still not fully understood very well. So try to understand all the keyword and all the experimental step. Therefore, when you attend your Viva, you can answer all the questions given by the examiner. Okay, right?
Okay, thank you, doctor.
Okay. So is there any other questions from the pros? Any other questions? Any other lecturers got any questions to Raad? All right. If there's no question or want to ask later, and so I will shut the presentation for Raad here. Thank you so much, Raad, and hope to see you soon in Viva soon. Okay, good luck. Okay, thank you.
Thank you, doctor.
Hello, doctor.
Okay, the next presenter is Sana, right? Okay, Sana, my student. Okay, I
Yes.
will leave. I will
Yes,
Hello,
pass it to-
doctor.
tunggu
Yeah,
Dr.
the
Venu.
next chairperson, please.
Tunggu Dr. Venu, kita akan start.
Dr. Vanu or Dr. Eric.
Sudah, examiner Dr. Venu. Dr. Venu and Dr. Eric.
So if maybe anyone, Dr. Eric or Dr. Vanu can be the chairperson or yeah.
Sorry, doctor. Can you see my slides?
Yes.
Okay,
can
thank you
see.
so much.
Okay, just wait.
I'm here already.
Just wait, Dr. Venu.
It's okay. I'm waiting, doctor.
Bob call Vino.
Saya dah WhatsApp dia kata kejap.
Oh, kejap, okay. So we wait for him lah kan? Ke kita nak continue dengan next presenter? Dr. Tashrik. Should tak ada. Okay lah, we just wait a few more minutes.
So Dr. Zainul dah masuk dah, Kos?
Dia dah, okay. So we continue with next presenter. Now I would like to invite Sana, student under supervision Dr. Heng, and your examiners, Dr. Eric and Dr. Venu. So you can start, Sana, if you're ready now.
Thank you so much, doctor. Thank you. Thank you. Hello, doctors. Hello, students. I'm Sana Yasin, and I'm under the supervision of Dr. Leo Hen. My title of my research project is about characterization and immunoactivity of fermented selenous linters, crude extract in macrophage cells. This is our final presentation. Sorry. This is my content. I will cover all the content here. First of all, it's my introduction and the background of my study. First of all, like my background of my study, since we are studying like selenous linters is medicinal mushroom. It came from the idea like people right now looking for something for immuno-enhancing products. So, but this is came because like aging population right now and increasing by WHO, they related that information. Also, the second one, prevalence of the infectious diseases without any antimicrobial thing. So it made it significant global interest in food and medicines that boost immunofunction, like nutraceutical or immunoceutical things, or any agent like helping us with the immunology. So we went to the natural product. Okay, so natural products, one of the most common one is medicinal mushroom, came from the Chinese traditional medicine. So medicinal mushroom, it's like, for example, like the one that's most common, reishi, mesitic, and metic, have been recognized by the people and in scientific, field by their boosting property for centuries. So this is all came from their bioactive compound, like beta-glucans and terpenoids, play a crucial role in enhancing immune responses and reducing inflammation based on the experimental results. So, sorry. This is my study species, Selenous linters, the wild one, and then it's common. It's known for its biological active, anti-cancer, and immunomodulation. And the second thing, we are looking for the bioactive compounds inside the Selenous linters. The most common one and the one like enhancing the biological activity is polysaccharide and terpenoid and phenol. And for example, why we are looking for the bioactive compounds? Because like inside each of the class of bioactive compounds, there is like, active compounds or key active compounds under this class of the compounds. For example, here we have histidine and inositol, one of the highest and strongest antioxidant activity of the phenols. They found it from Selenous linters, wild one. So what's the benefit of finding this one? So it's a benefit for the application in natural immunomodulatory agents. There's a new field, it's called immunoceutical, like people can get it without any side effect and toxicity. And the second one, potential for clinical application, for example, like in its application, for example, like in liver toxicity, any kind of the application. And third one, for drug development, fundamental units. So the focus of my study is about immunoactivity of selenous linters, the fermented one. So I did a literature review about how it's going on. So the first one, humoral and cell-mediated immunity with the adaptive one, and we have innate immunity. My focus is going to be innate immunity, but I just like studied all of them. So first of all, we have T cells. Like the effect of the selenous linters extract on the T cells enhancing cytokine production, interferon gamma and interleukin-4, and Th1, Th2 balance the cells, and also lymphocyte proliferation, like splenocytes. Most of the studies have been done in the in vivo and the mice. So the second one, B cell, also like its effect on the B cells by the proliferation of the B cells, and also augment antibody production. So two main of the function of the B cells. And the other side, it's macrophage. For the macrophage cells, it's affected like by macrophage activation and polarization. We know like macrophage after the activation is going to be M1 and M2 classical, and the other one, alternative one. And then we have phagocytic activity of the selenous. It's enhancing by the polysaccharide or any compounds like extracted from selenous linters. And the other one, nitric oxide production, also is one of them responses by the macrophages, like for the any inverted like microbes or any kind of the immune system facing the like fight, like any kind of the responses. So, and the other one, cytokine production. So I mentioned like my sample is going to be fermented selenous linkers. So my sample is going to be a new type of the fermentation innovation of the fungal processing pathway. This idea came from after like selenous linkers. It grows on mulberry tree, and it's so hard to collect that mushroom, and also like it is grown like in between 2-3 years, and the fruiting body of that mushroom, it's hardly people can get it. Most of the products in the like the markets and everywhere, like medicinal market, the common one like Chinese medicinal market, most of them it came from the fermentation or processing, like downstream processing by the submerged fermentation. The products all produced by the mycelial growth, not the fruiting body, not the whole one. So the newest fermentation type, they call it, there's like two of the new ones, submerged fermentation and the solid state fermentation. Submerged one, I'm not like going down to it because it's not like my focus, but I just want to explain that part. Submerged one, it needs water, and the mycelia will grow and release all the metabolites inside the water, and you have to feed the water. But the other one, solid state fermentation, is the same as how the naturally growing the mushrooms on the like that tree, mulberry tree, or some places. So that one, solid state fermentation, they are using substrates like brown rice and so many of the substrates, but the brown rice is the most common one. They will put the mushroom with it, and then the mushroom will grow with the substrate. And then this is like the idea. It's the cultivations and processing of fungi, addressing the community needs and expanding its application, because we are putting substrate. So it means like as the hypothesis, it's gonna be enhancing the releasing the metabolites more. So there is they have done like 3 studies on the solid state fermented selenous linkers effect on the vital state. The first one is showed like anti-inflammatory activity, and the other one anti-inflammatory effect, and the other one anti-inflammatory. Both of them anti-inflammatory, but in different one. The one like on the macrophage cell, and the other one, the third one, is on the animal-based in vivo one. So they have done like in 2009, 2010, 2014, and from that time there is no other study, and also there is no other study about the characterization and what's like what is the active compounds, and what's the characterization of the crude polysaccharide. So problem statement of my study is going to be the immunoactivity of fermented selenous linkers extracts in macrophage cell line with the determined extract or crude extract, because it lets the research to provide scientific background of the effect of fermentation on bioactive compounds. So my research question and hypothesis, the first one, how like fermented selenous linkers gonna by hot water extraction influence the antioxidant activity and nitric oxide production and the transcriptional profile of the pro-inflammatory cytokine production in the treated or differentiated THP1 cells. As we said, like this one is fermented, there is like no evidence, there is no background, scientific background about the outcome, how like the fermented one is it working the same as the wild one? So the hypothesis of this study, the polysaccharide from this one, the whole solid fermented selenous linkers, it's gonna be as wild type, so significantly modulate human macrophage cell line, THP1 human immunoresponse. Also, like most of the study have been done on the murine macrophage, not the THP1, not the human monocytes. So this is I have 5 specific objectives. The first one, characterization of the bioactive compound. Second one, determining the antioxidant activity of the crude polysaccharide, and then determine the extract from third, fourth, and 5. It depends on the cell culture. Determines the cytotoxicity and cell proliferative effect on the differentiated THP cell, and also investigate the pro-inflammatory cytokine gene expression on THP1 cell after treating with the polysaccharide. evaluating the nitric oxide NO production in the treated macrophage cells. My methodology, the main methodology is depending on the hot water extraction, the one like naturally have been used back of the day. This is my mushroom. I got it like powder because mine it was polyplate fermented, but I just put it in the picture. So fine powdered fermented spirulina centers, and then by hot water extraction, the ratio 1 to 14 for 4 hours, like at 80 degrees Celsius. And then purification by the 95% of the ethanol purification. Yeah, the simplest pathway with the extraction, because this one people also like outside of the lab they can use it. Okay, then air dried crude polysaccharide, crude polysaccharide, then go under the experiment. The first one I have done my the first method. It's small system identified for my spirulina centers to be sure like the sample the species they have used it is spirulina centers, because as the morphology of the spirulina centers, most of the spirulina centers, and there is a hung, there's another species, and under the different genus, it will look like the same. So it needs the moisture system identification to make sure like your sample is a spirulina center. And then phytochemical characterization due to the colorimetric assay. I have done like polysaccharide, beta-glucan, tretinoin, total phenol, total flavonoid in triple sample, duplicate, and then antioxidant activity, the free type with the antioxidant activity test, DTPS, FRIP, FRAT, and ABCS. And the last part of my research, my methodology is based on the cell culture. We are like dealing with the TFM531 cells in the lab, and also like goes under the MTS assay, natural oxide production, and then after the treatment, we have to do the gene expression by real-time PCR. This is my research flow chart. Crude polysaccharide extraction, the main part of my job. I have done the characterization of my crude polysaccharide, and then goes with the in vitro immunoassay, TFM1 cell line. My result and discussion. First of all, my study species sample DNA identification. I visit ITS 4 and number 4 and number 1 for my primer to identify the strength of my sample, spirulina centers, and the other species like I use it as a product control. After I've got my bonds, after the PCR product, and I use the ITS 1, ITS 4 product control. Also, like why I use it, there's two jealous for it, because the first time I didn't get my band for my spirulina centers, and then we tried again, we got it, and also like we use a different primer, but I will explain that thing. So, but at the end, I got it my results for my spirulina centers with the ITS 1 and ITS 4, not the new primer. That's why I didn't put my data here. So then after the DNA sequencing, we send it for sequencing. The data, the raw data, we got the raw data, and then I did the blasting for my data, and then the ID score covering, then finding the with the highest score, and then I did the both system, phylogenetic ID identification for fungi. This is my result. So the first one, I sampled spirulina centers belongs to that strain, and this is the NCBI ID, gene bank information, and the similarity score was 98, and the EVA value was 0, and the rest was I used it as a product control from IPS region. So the genus Rhinas, Grifola, Sweden, and I'm not familiar with reading their name, Pleurotus irrigine. Okay, first of my objectives that I covered in my work, characterization of crude polysaccharide, the bioactive component. I did the total carbohydrate content, total phenolic content, total sericinoid content, and total flavonoid content. I did the hot water extraction. My study is all about hot water extraction. After I got my results, the total carbohydrate was 132.8. It was high, and the second one I got total phenolic. It was not that much high, not that much like low, because when comparing with the wild one or the other species of the medicinal mushroom, it was like low, and total tritiptany 1.19, and I didn't get total flavonoid because I didn't get total flavonoid, and there is a paper saying like there's a total flavonoid in the wild one, so I did another tried another method for my confirmation and comparing. So I did the ethanol extraction. So I got at the same, I got no total flavonoid. So then I just stayed with this result. So in my sample, I don't have total flavonoid. But the good thing, I got total, total certified like hair with ethanol. But for the total polysaccharide, because my focus is going to be polysaccharide or because amino activity belongs to the polysaccharide or the beta glucan. So that one with the food water, it was threefold higher. This is a diagram of my comparison. Only the polysaccharide was higher in the ethanol extraction. This is due to the structure and molecular structure of the organic of the treatment. The second one, after I got my polysaccharide the higher level, so I did the glucan content. The main component in the total carbohydrate is glucan that have two types, alpha and beta. So I did the experiment for them and contribute to the studies, the immunomodulatory effect and antioxidant effect mostly come with the beta glucan. So this is my result of the beta glucan. Total one, it was 56, more than the standard kit, and also only I got a little high level of alpha glucan, and also the beta glucan is 36 percentage. This is the diagram of my glucan content. And here is comparison between beta glucan percentage in the control beta glucan and my fermented selenium test. So it was significantly different between them. Determine the antioxidant activity. My second objective, determine the antioxidant activity of fermented selenium test in polysaccharide. So here is my IC50 values of mg per ml of ascorbic acid, fermented selenium test due to the radical scavenging activity. The first one, I didn't get IC50 for due to the low amount of the compound responsible for antioxidant activity. Only the froth gives the result for the IC50. Here's the diagram of the froth. So it was more than 50%. It can do the reducing activity, reducing ability for the crop. The antioxidant inside my sample, it could reduce the crop, reducing Fe 3 plus to Fe plus 2. So the result, it shows the less potent antioxidant activity of sample compared to the standard, but still there is the antioxidant activity. So antioxidant activity for the DTPH and also FDS. So I didn't get the IC15, but also their activity, it was 34.9 for DTPH and also 47% inhibition in the FDS. While this value don't allow for the determination of the IC15, but also indicate that there is effectively scavenging activity, small portion for both of the DTPH and FDS radicals at the tested concentration that I used 10 mg per m, my highest dilution sample. To conclude my characterization and antioxidant activity results, first of all, the extraction, when you are dealing with the extraction is significantly the method that we are using affect the composition and yield, and with extraction yield higher total carbohydrate, because I'm focusing with the polysaccharide, so for me it was better, but for terpenoid, it was ethanol extraction was the yield better amount, and the polysaccharide, the one that it's giving you the result, the higher one, it's better to be characterized by structure composed. Characterize it, and the structure composed of beta glycosidic linkage. It means beta this type of linkage. It gives the it's related to the immunomodulation and antioxidant activity of that compound. Impact of the phenolic content and antioxidant activity. After I compared my results with the other papers, so my phenolic, it wasn't that much low, but didn't give them because the amount of the phenolic in mushroom extract directly influences their radical scavenging. So why mine it wasn't? Then I forgot to cite this one. Then the antioxidant activity, sometimes it's not depend to the amount of the total phenolic. It depends to the specific compound. For example, inositol, one of the strongest antioxidant activity in the cholinesterase. So flavonoid content in the wild cholinesterase. In Thailand, they have done the research, so they found that there is no, the only species of the Philinus genus, Philinus doesn't have flavonoid. The characterization result is variable among the studies that have been done because of the temperature extraction method and ratio of the dry powder to the solvent and type of the solvent. This made my result interpretation hard to compare with the other paper because each of the papers was everything was variable and everything was different, so it wasn't that much giving the significant explaining. But my job, and this is my reference, and this is thank you so much. Just let me to go back to my slide because my method is contain cell culture. So, but my working is just staying here because since I started, I'm just working on my characterization and working with the cells. Right now, our cells we didn't proceed with our cells to treating it because of the instability of the cell growth and low growth rate of our cells and struggling with the cell culturing. So we are figuring out our cell culture. That's why I don't have any data about cell culturing. Thank you so much for your listening.
Thank you so much, Sana, for your good work. I believe you have done a lot of work and nice presentation, a nice slide, comprehensive presentation. So I'd like to congratulate you on completing everything, and congratulations to your supervisor too, Dr. Herb, for supervising good student and good project.
Thank you so much.
Okay, now let's go for Q&A session. Now I would like to invite Dr. Eric and Dr. Venu for any questions to Sana.
Dr. Eric first, can?
Yes,
Okay.
Okay, thank you. Can you go back to objective? Can I see the objective
Yes.
again?
Yes, sir. Let me to type down.
So your work,
Yes.
okay, so your
Yes,
work is complete already or what?
No, doctor. First of all, I didn't know our final presentation is gonna be in November because we were discussing with my supervisor. Our work with the the other three objectives is gonna be on November, but then we just knew that thing, so we just present the result that I got it because mostly I was busy with my characterization. And then I did two methods to just get my result done.
So the experiment you carry out is you done the THP1, the macrophage cell assay, the cell.
No,
You
that's
done?
only growing because we are teamwork. We are working in team. So with my seniors, we are dealing with the cells, kidding and packaging. But I don't know if you are aware of that thing. Sometimes nowadays, we are facing some of the low growth and instability of the cell growth. This is the problem that we are facing right now.
Okay, because how many months or how many days you left before you have to submit the final thesis?
I think that's less than month. This is just less than month.
December.
The 8th of December. I don't know, it's short time, and also as a mixed
maybe 2 months left, maybe about 1 to 2 months left, I think. So I think maybe you my advice is actually if you cannot complete some of the experiment here, maybe you should think just cut off the objective because you have 5 objective here, It's
Yes,
considered quite a lot for the mixed sport student. So maybe you can cut to 3. My suggestion this you have to discuss with your supervisor.
Okay,
because
doctor.
no point. I think you don't have time. If you don't have time to finish up the experiment, better you just reduce the objective of this. Maybe just reduce to 3. The fourth, I think, because if we go back to slide number 17,
Yes, doctor.
17, 17.
Okay, doctor. Yes, doctor.
Here, you see, because here is a ascorbic acid is a
Yes,
control, right? It's a
yes,
control, right?
yes, doctor.
And one thing you notice or not, you want to do the antioxidant DPPH, you don't get the reading. Okay,
Yes, yes,
ABTS,
doctor.
you only get the reading of FRAP actually. Is this
Yes,
good
doctor.
FRAP readings compared to the control?
No, doctor. No, doctor.
So this my question, because if this is not, because usually standard lah for oxidants test is DPPH lah. That's the most common people use for antioxidant, right? That's common
Yes.
lah. Actually, that's a common. Why you didn't get the result for this one? No result, or is unable
No,
to determine, or is no result?
unable. No, doctor. Unable to determine. Even here,
Unable to determine. Okay.
let me to show you the. I have my table on that one. I just like put here. The DTPH it was like 34.7 because I only did
Okay.
for hot water extraction. So maybe due to the low amount of the phenolic,
Maybe
because
you should
phenolic
put this
and
one,
sunlight.
You should put
Okay.
this one instead of the not able to determine, because at least this has got some result. Look
I
back
put
on
it.
you, because remember your test sample is on the this one, the fermented what linters, not the ascorbic acid. Is look back on the thing, how you present the things. It's
Yes,
better
doctor.
you got result, but maybe it's not as good as the one, but it's better to put the last slide that you show. At least you show some result on it. If not, it's just blank, right? It's just everything's blank, nothing to show. Actually, look bad on you,
I just
because
like.
your DPPH is not one to show on the ascorbic acid, because that's standard lah, vitamin C, right?
Yes,
Nothing to tell people to do it, but your one is actually, I don't know, it's good or bad, because you want to show you got activity is good, or you don't want to show activity is good. I don't know. It's based on the interpretation, but better don't show the ND.
Okay.
Not able to determine. It's not good on your things. You should show you've done some result, but is the result maybe is not living to the standard or this kind of thing. At least show, but not put on the ND, because remember, this is your experiment. We
Yes.
are not doing ascorbic acid. Is you do the fermented one actually. At least you put some result, not ND. At least.
Yeah.
So I think
Thank you so
the
much, doctor.
next slide.
Okay. I put it here. I don't know because
This
I
one.
didn't want to.
This one same also. I think you better discuss. I think
Yes, I
now
discussed it
actually,
here.
if you can finish out the other experiment you do, yes. If not, I think better just cut off. We write or do some more discussion, because I don't think you can finish the cell one. I'm not sure. Are you doing currently the cell line already? No.
No, just like observing. Like just
See?
passaging the cell, but the growth is too low. The
But if
complaint
you
is
can,
not.
you finish it lah. If not, better you discuss with supervisor. Maybe just limited to your objective, so you don't do bad, because you cannot complete. No point. When you submit the thesis, you need to complete the thesis.
Yes,
I think you only have maybe 1 month only left. I think I think December you have to submit already. I'm not mistaken. December, right? Early
Yes,
December or mid
the first week of December.
You see, you only have 1 month left only.
Yeah, yeah.
So don't be too ambitious lah. I think because people understand you're a mixed school student. Discuss with supervisor. Dr. Heng should be understand this kind of problem. I
Yeah,
think
of course.
discuss the thing. Not put ND because ND look like you don't do anything at all. Put the experiment is some result is okay. Don't put ND. ND is look bad on you. And discuss the thing. If can do, can finish. If you cannot, because on December you have to submit the final copy already. You don't have time. You only maybe you need you start writing already. I hope you writing part of the Yeah, I
Yeah,
hope
yeah.
you're writing the introduction, the methodology, the research you know, try to have it out, okay?
Yes, thank
That's
you
all
so
my
much.
question. Because you put too many objective after that, I finish already. Then the other part I don't see
Thank you
anything.
so much, Doctor.
Okay, that's all my question. Thank you, Chairman.
Thank
Chairperson.
Thank you.
Thank you, Dr. Eric. Okay, now Dr. Venu, you have interested.
Yeah. Hi, Sana.
Hello, Doctor. Hi.
Yeah. Your presentation is good, like what Dr. Shaida said, but I had the same query as what Dr. Eric have mentioned.
Yes,
Okay, you have so much of objective, but certain objective you couldn't complete.
yes,
Okay,
Doctor.
so the objective that you can't complete, you must omit it and just show that whatever that you have completed. Okay, so like
Good, Doctor.
showing ND data, so not determined
Oh.
is not
Good.
good, like what Dr. Eric said. It's not
Yeah,
good.
thank you so
And
much because this is my first, research presentation. Maybe that's why I didn't know that thing. Thank you so much. It's
yeah,
so
yeah,
good for me. Yeah, this is a good point. Thank
yeah.
you.
So when you have 3 objectives, you have completed. I think the first 3 objectives you have completed, right?
Yeah, most only the one like related with the cell, but the problem is just like things going on. It's unexpected one. That's why, and I did 2 methods for confirming my results too. Busy with the like characterization. That's why. Sorry, let me go back to my. Here's my objective.
Yeah.
2 of them is finished. Only like the third one is depending on the cell.
So I think so your 4th objective you have not done anything yet.
No, because of the cell condition.
Yeah. So I think you can just complete your 1, 2, 3. Your 3rd objective you complete it, and then you just write down and remove 4th and 5th objective lah. Because if you don't complete, you put, then there's no data for it, and it is indicating that you have didn't done your work.
I see. I will discuss that in the
Yeah.
next supervisor then.
Yeah,
Thank you
yeah.
so
You,
much,
you need
Doctor.
to you need to talk. Yeah, you need to talk with Dr. Heng. I think Dr. Heng also understands that.
Yeah, of course.
So yeah, so you don't have to worry because you don't have to finish 5 objective. You are just mixed mode. Okay, everyone, you even the examiner understands that. But when you submit your thesis, there must be some data to show that you have completed the objective that it is been mentioned there.
Understood, Doctor. Thank you.
Okay. Yeah, I had the same when I was looking through. I had the same thing with Dr. Eric. So
yeah.
that is the main part that you have to get it done and finish your objective 3, at least your cytotoxicity work.
Yeah, I see. Thank you so much, Dr. Vanyo. Thank you so much.
Yeah.
Appreciate
That's
it.
all for me, Dr.
Sana,
Shaida.
Yes,
Sana, I
Doctor.
want. Yeah, can you go back to the next slide?
Next
Next slide
slide,
with
Doctor.
the
This one?
ethanol and the hot water extraction.
Okay, Doctor.
One. Okay.
This one, Doctor.
This one, yes. This is I think it's the carbohydrate compound is higher, right? They have how many time? At least four
Three
time higher than the ethanol extract. Yeah lah,
Yes,
but
Doctor.
the tripidins is almost lower lah compared to, at least it's got expression on the. So this one I think you should highlight the way how the result different, and what the purpose what you use, because you use hot water, right, and ethanol, this kind of extract. I think you should, when you do discussion, you should highlight what the purpose of this 2 different type of method of extraction, hot water or ethanol. Let's say you want a different type of content inside the extraction, right? So let's say you want the tripidins, triterpenes in higher 3 times lah. It's nothing like can say, but it's at least you can highlight the differences between the carbohydrate is what? Is glucose? Nothing,
Yes,
right?
Doctor.
Phenolics,
Yes.
nothing.
It's polysaccharide.
Glucose, nothing, right? It's not affected on the phenolics also. So in the next biotic compound, people usually don't discuss about carbohydrate for extraction, right? For compound, right? That's usually yeah, you have to usually people with phenolics or these kind of things. This is the better result, double or twofold compared to the ethanol extractions. No. Are you compare the other experiment? Are you compare the hot water and ethanol extract in the next
Doctor,
experiment?
no, can I just explain this one? The ethanol extraction, I have done this one for myself and for my literature, because like in the literature when you are like doing grading, they are saying like there is a phenol, like there is flavonoid, there is these trace noise. But when you are doing in hot water extraction, maybe you are not getting things. So that's why when I didn't have my flavonoid, so I just wanted to make sure like I will have my flavonoid by the ethanol extraction to just like discuss that part, like by ethanol, the solvent organic one, so I can get my flavonoid, and there's flavonoid that cannot be detected by the hot water extraction. So I did this part only for confirming my results and make sure like about some of the papers, but like some of the papers like mention something, the results saying something, but when you are like running it, the results not the same. So one of the problem of the that's why like Dr. Herring he chose this project, because there is no standardized results and calculation and the background of the characterization. Whenever you are doing, when I'm comparing my results to a paper thing, so so many things it's not matching. So
Okay.
for example, this one, yeah.
Yeah.
Thank
Okay,
you.
understand. Yeah, I understand, but you need to highlight the because like I say like not much result inside your finding as the moment. You need to highlight for discussion as the thing. I'm not sure lah. Maybe you can compare with the other researcher data or this kind of thing just to highlight any differences. If not, very difficult lah. I know it's quite difficult to write on the discussion on other things. If not, how you gonna present it because you don't have really much data. Anyway, that's all my question. Good luck, Sana.
Thank you so much, Doctor. Thank you so much.
All right, thank you so much, Dr. Eric and Dr. Venu, and thank you, Sana, for your presentation, and all the best to you.
Thank you so
for
much,
your
Doctor.
thesis. Yeah, okay. So I pass the session to next chairperson. Apa?
Yeah, I'm here.
Oh yes, Dr. Ong. All right, thank you.
Thanks, Dr. Shaida. All right. I checked the list. Jingtao is around, and then Dr. Tashri, are you around?
Yes, I'm coming.
Okay, great. Thank you. And then Dr. Eugene. Dr. Eugene.
I think we need to wait a minute for Dr. Eugene. She knows already.
Okay. Yes. So we wait for Dr. Eugene. Thank you, Dr. Chong.
Maybe Jintao, you can share your slides now. Dr. Eugene said he's waiting for WebEx load, so I think he'll be joining us in 1 minute.
Okay, great. Hi, good morning. I'm joining by phone because I do not know why my PC cannot join in. I'm okay now. Thanks. Okay, thanks, Dr. Yuji. Okay, so everybody is here. So the third presenter will be Jing Tao Pan, student supervised by Associate Professor Dr. Chung Yi Siu. Jing Tao, you may start now. Please go ahead.
Morning, everyone. My name is Jin Tao Pan, and my supervisor is Dr. Zhang. And my topic is pentapeptide identification for the inhibition between caligulin B S100A9 proteins by in silico approaches. It's my context. In introduction, from 2060 to 2033, there are 370 new drugs was approved by FDA. The number of peptides and oligos occupied almost 10% of new drugs. Despite the rise of small molecule drugs in 7 years, peptides still occupy an important position. Short peptides are cost-effective products with high purity and more bio-friendly as bicycle biology molecules. However, they have some limitation, such as short half-life and low overall bioavailability. And we have response methods such as terminal protection and using unnatural amino acids. So there are so many peptides used in skincare and some ocular disease, such as CMV and dry eye. And in the mouse eyes, the intravitreal half-life of peptides is known to be in the range of a few hours. So the peptide drugs is important, but how can we design or screen the treatment peptides? There are two ways to build peptide libraries. First is the chemical method, SPPS, involves sequentially adding protected amino acids to end of a growing peptide chain, ushered to an insoluble resin with raising steps to remove excess regions and deprotect the peptide chains. Second is biology methods, to produce DNA library first and then putting them into phage for merging with phage's DNA, coding core proteins. Those core proteins contains our potential peptides, so we can easily using targeting receptors to select our treatment peptides. Why targeting S109? But those methods rely on random selection and extensive preparation, and subsequent preservation level must be expanded. So virtual screening wherein silico approaches demonstrate that they are less difficult to use, process a high throughput screening, and are economically efficient in peptide development for drugs. Apart from that, the result of virtual screening offering 3D interaction information. So why we choose the S100A9 for targeting? S100A9 monomers looks like M-shaped and is easily assembly to amylose, not only self-assembly, but also assembly with other members of S100 family. And the S100A9 amylose cause lots of disease, retinal toxicity, AD, and cancers. Amylose structure of S100A9 is involved in regional dysfunction, and S100A protein as a molecular targets in the ocular surface inflammation disease, and is the S100A9 amylose is the attachment core of A beta fiber for progress of AD. They have different kinds of the structure can be hydrophobic, templating, coating, and losses interaction with A beta fibers. And this picture shows the S100A9 on the surface of A beta fibers. Yes, it's how to screen a tree peptide. So there are three S100A9 inhibitors can block amylo aggregation of S100A9. ApoE appears to be used to clap S100A9 homodimer in solution and restrict its extension. And this article mentions from less than 50 to less than 54 is the connect area between both ends of each S100A9 homodimer in S100 fibers. It's worth mentioning that only OLEA, a natural killer chemical in olive oil, has the ability to disaggregate S100A9 fibers. About the pivot stick between tryptophan 88 and hydrogen bonds with adenine 85 and glutamate 52 of S100A9. Consider the cost of peptide production and the number of screening. The number of peptides increase 20-fold when each amino acid is added. So we use pentapeptide library to screening the treatment peptide, which can targeting those active area already be published. Hopefully one day after validation, the pentapeptide tree ability, the pentapeptide drugs can load it on contact license for long-term treatment, and maybe it can use to treat the AD disease delivered from eyes. Is my program statement. S100A9 self-assembly is relevant with many diseases, and to block S100A9 self-assembly is required to identify novel peptide drugs that are more biological friendly and have stronger binding ability. And build a virtual peptide library makes it simple to uncover prospective medication for treating not only S100A9 assembly, but also additional disorders in future. Research aims and objective. First, to construct a pentapeptide library for virtual screening. Second, to identify the pentapeptides that can bind with S100A9. Third, to evaluate the interaction between pentapeptides and S100A9. Is my methodology. Some 3D structure of molecules will be downloaded from the protein bank and ChemiSpider database. OLEA will be my positive control to compare the binding ability of peptides. Producing the pentapeptide library via RDKit, a Python API, it can transform the peptide sequence to PDB format files. And the OpenBaba will be used in converting the PDB to PDBQT, because the PDBQT is the spatial format used in Autodoc. Binding free energy and hydrogen bond of Autodoc results will be analyzed and ranked. The most detailed analysis will be used the AMBER 2080 to do molecular dynamic simulation. And the Python will use in batch process and manage the data. There are my results and discussion. In recent research, I use arrangement and combine of 20 animal acids in 2 sites, resulting to 400 results, and then putting codes into the 400 results. We can produce any peptide library when change the codes putting in the middle. For example, we put the same 40 results into the middle of the 400 results, can generate the tetrapeptide library, including the 160,000 peptides. And the pentapeptide library can be achieved by putting the 20 amino acids into middle of each peptide in tertiary peptide library. 20 multiply 160,000 equal to 3,200,000. And using RDT's API 10 days to build the pentapeptide library. The size of PDB library is 28.7 GB. And then we convert to PDBQT format. Next is we use Autodock to docking 300 times between ODA with S100A9 to reproduce the docking results. And the binding free energy is negative 8.23 kcal per mole. Our result is similar with the results of articles. And then we meet a big problem. We don't have enough resource to docking all pentapeptide library. So we docking the tripeptide library with S100A9 first, because the tripeptide library only 8,000 peptides, and then choose the tripeptides with high score of binding free energy as code to isolate the pentapeptides. Using those pentapeptides docking with S100A9 to screen the potential treatment peptides. Is the docking results of tripeptide library. Tripeptides exhibition binding free energy at negative 8.0 kcal per mole, which is close to ODA's binding free energy. So we choose the three tripeptide as code to isolate the pentapeptide library. Because pentapeptides with WWF and WPW codes are more likely to provide a large number of outcomes with lower binding free energy. Especially those pentapeptides include the WPW code. There are two docking results lower than negative 9 kcal per mole. The WPWTY with the best binding free energy binds outside of the S100A9's active site, so we incorporate the WWPWH with the second best binding free energy into the MD simulation process. And the other one, the TWYW peptide binding. Sorry, Lena, can you?
Sana, please switch off your mic. Please unmute.
Sana.
Thank you. Okay, sorry for that. Jingtao, please continue.
The WPWY peptide with binding free energy negative 8.7 also was chosen because it has the greatest number of hydrogen bonds with S100A9. There are 7. So we successfully identified 2 pentapeptides with potential to inhibit S100A9 assembly. Their binding free energy is lower than ODA and have more interaction with S100A9. And then we saw 100 nanoseconds MD simulation 3 times to discover more details of their interaction with S100A9. It's the MSD data. 2 peptides binding more strongly to S100A9 than ODA because the MSD represents the distance between ligand and the receptor. If the ligands move too far apart, they may break away from the receptor. And the MSD curves from 3 times MD simulation of ODA and S189 are unstable. You can see the black, they are unstable. Next is the RMSF. RMSF measures the average atomic mobility during molecular dynamic simulation. In other words, the lower value in this residual size means this residual more stable during MD simulation. And especially the results of the second MD simulation, the WWP-WH peptide shows a more stable binding ability with S189 than the WPW-YW peptide. And the ODA only in the third time MD simulation showed the better MSF results than both peptides. However, MSF data shows that 3 compounds have a similar propensity to bind to S189. This data shows the ODA generates less hydrogen bonds with S189 than the 2 peptides. You can see the number of hydrogen bonds of ODA can create with S189 is less than 6. And WWPWH may generate up to 11 hydrogen bonds with S189 on occasion. Is the number of the hydrogen bonds between S189 residues and ligands during the 3 times MD simulation, and the hydrogen bonds number is less than 1,000 were not sure. And the y-axis is the exponential axis. You can see it's the 100 and 1,000 and 10,000 and 100,000. So compared with that of ODA, both pentapeptides have wide range of the hydrogen bond interaction with S189. From glutamic 52 to aspartic 65 and lysine 93 to proline 107. And this is MMGBS results. It's a pro-minor approach for determining the binding free energy of ligands to proteins. Binding free energy results suggest that 2 peptides combine stronger with S189 than ODA. During MD simulation, ODA's total binding free energy was 5% greater and near 600 kcal per mole than the 2 peptides, and the difference is highly statistically significant. Then we focus on the contribution of individual residues. The hotspot residues contribute significantly to the total binding free energy of the complex, topically lower than negative 1.0 kcal per mole. In comparison to ODA, the binding free energy of 2 peptides not only is contributed by the potential active sites from agony 55 to alanine 89, but also by other active sites from leucine 49 to valine 58 reported in some articles. And this the data indicates that the 2 peptides can have additional interaction based on ODA's interaction with S189. And the data also overlap with number of hydrogen bonds in S189 residual results. 52 to 65 and 93 to 107. Okay, is the conclusion. And we successfully constructed the entire pentapeptide library, and it was released on USM library, and then screened the possible therapy pentapeptides and assess their binding ability with S109, which is superior to the natural products only. And in comparison to prevention inhibitors, the two peptides will interact with S189 residues from alanine 85 to alanine 89 and leucine 49 to valine 58, indicating the significant therapeutic potential. And recommendation for further research, the two pentapeptides should be evaluated and verified in one experiment to demonstrate the potential to degrade S189 fibers while also inhibiting the elongation of exciting S189 fibers. And then we can use some animal models with an AMD will be employed for in vivo invalidation of two peptides like amino can solve the regional toxicity caused by S109 fibers. Okay, this is my final presentation, my flowchart and Gantt chart and reference. That's all. Thank you for your time.
Okay, thank you, Ching Pao. I would like to invite Dr. Ashrik to ask questions or and give the comments. Dr. Ashrik.
Okay, thank you, Prof. Ong. Okay, hi, Jing Tao Pan. First, I would like congratulations for finish your research work. Okay, I think that's a hard work lah. All right, can I have few question? Okay, if we look back on your page 32, okay, if you go back for your page 32, okay, and I think also page 31 of, yeah, from 31 until 32 lah. So you talk about hydrogen bonds in these interactions, okay, between the peptides and also the calnogorine. Okay, my question, why you only choose the hydrogen bond for the analysis and why not the others bond like hydrophobic, like maybe salt bridge or ionic interaction, so on? Why only hydrogen bonds in your study?
About this question, first, in these are articles about the inhibition of the S109 amylose. They only mentioned the peptide staking and hydrogen bonds interaction with S109. And the second is hydrogen bonds is an easy interaction we can access. Yeah.
Okay, meaning the other interaction like hydrophobic we cannot analyze using this any software because you have the all atomic trajectory, right? So
yeah, but actually they including all of the interaction such as in this the binding free energy resources. Yes, like one vita and something else. It can access all of the interaction, but for easy to show to others, I think the hydrogen bond is we can directly see what happened during the interaction.
Okay, all right, and then okay, so you're using MMGBSA, right? Molecular mechanic generalized bond, MMGBSA surface area lah. Okay, so can you mention me others than MMGBSA that usually the researcher use other than MMGBSA? What else that researcher use?
What
other
are they?
than MMGBSA? I think we have another one more. If protein-protein interaction other than MMGBSA, we have one more.
What you mean? I now get your point.
Okay. In your study, you're using MMGBSA, molecular mechanic with generalized bond and surface area. But other than this analysis, what else that usually the researcher use for the protein-protein interaction analysis?
Actually, they only consider the binding free energy actually.
Yeah. Binary free energy also can be used by this analyzer MMGBSA, and then we have another thing, another analyzer MMPBSA, not G, not B, but P, Poison Boltzmann.
I know.
Okay. Okay, Poison Boltzmann. So maybe you can try to find the difference lah after this. Why you using GBSA and why not PBSA, and what the different and what is others lah. Okay.
Yeah.
Alright.
I think different is about the solution, and I can remember all of the things I read it before. Yeah.
Yeah. Okay. But it's not. But actually, if you have the trajectory of your MD simulation, you also can analyze by MMPBSA. But MMPBSA, I think it need a huge power of computer in order to get the value. But actually, this analysis more suitable lah for the protein-protein interaction because your peptide is protein. Okay, usually MMGBSA we're using for the protein ligand, but it also can be used by for protein-protein interaction, no problem. All right, I think that's all first from me. Maybe we can pass to Dr. Eugene for the next question. Prof.
Okay, thank you, Dr. Tashrik. Dr. Eugene? Yes, thanks, Dr. Tashrik. So maybe I'll just start with a simple question. In your conclusion, you mentioned something about the USM library. Can you explain a little bit more about that? Release on USM library. So what does this mean?
Because we release out of the whole pentapeptide library, and we release on the USM library, so everyone can download from USM library online.
Oh, so the peptide library can be used for maybe screening for other projects and other researchers have access to it. Is that correct? Oh, okay. And then from here we can see the two pentapeptides that you have found contains the residues which are tryptophan and also tyrosine and histidine, which all have rings, right? Have some kind of ring structure, aromatic rings.
Yeah.
So do you have any sort of hypothesis or explanation why you think that peptides with these aromatic side chains is attracted or can bind well to the target?
Yeah, I think to holding the stable combined with S109, the peptide stick, you can see that the tridobone ATA and this one, and so many hydrogen bonds interaction. I think both of them can hold the stable interaction between ligands and receptors.
Oh, okay. So does it mean that if you have other variations of this, you will also bind well? Perhaps inside your
I'm not
library,
sure.
there are
I cannot
there's
say if
already
you
some
have
kind of
so many
combination, right?
rings and then you can bind in better. For my results, I think the peptide stick is a very important thing for the stability. Even in the tree peptide library, you can see that? No, this way. Sorry.
Tree peptide library.
Yeah, you can see the pipette sticking,
yeah.
pipette sticking, pipette
Yes, yes.
sticking.
Ah,
And
okay.
also the ODA, their pipette sticking is also important. So maybe
Oh,
it's the reason why only the ODA can disagree the oh, only ODA can disagree the performance of S109 fibers. Another one just stop EU prolongation, but ODA can disaggregate.
okay. So OLA also has tryptophan
No,
or
it's
aromatic
natural.
aromatics chain, is it? OLA.
It's a
OLA
natural
has
product. I'm not sure it's
natural product. I see.
yeah.
Okay, so it's not
It's
a protein
small
or peptide,
molecule maybe.
but it has some kind of aromatic or ring structure in the
Yeah.
compound. Okay, so
And in
that's
this
good.
article, they mentioned the peptide staking is very important.
So okay. So that's why the screening were able to select similar kind of structures with some side chain with this kind of ring, right?
Yeah.
Oh, okay. Yeah. So from the three peptides and then the pentapeptides, the free energy binding is better with the penta one. Did you compare, for example, between the peptide and pentapeptide
Yeah,
free energy
it's
binding?
better because you
Oh,
can
okay.
see only actually the tripeptide cannot achieve the negative A, and you can see that so many pentapeptide can achieve the negative A.
Oh, okay. So maybe other residues provide some kind of stability, even though the ring side chain is important for binding to the, what do you call that? Your protein is granulin B, but then supporting residues is also important to make it stable in the binding.
Well, how to say? Because I don't screen all the whole pentapeptide library. I screen the pentapeptide library based on my tripeptides results. So they already include many rings.
Okay, I
So I just
understand.
put
Okay.
amino acid in the I just put the tripeptide in the middle and the two sides.
Okay.
So yeah.
Okay. Yeah, it's interesting for me why this can happen. So in terms of the biological activity of this protein, you mentioned earlier that is it related to, in the introduction, is it related to amyloid beta?
Yes.
Is it
Most of
related
study
to Alzheimer's amyloid, or is it a different protein?
Because S100A9 is inflammatory factor. So you have the inflammatory first, and then you will and then you will cause the Alzheimer's disease. So many researchers think the S100A9 amylose is the attention core because they have the core first, and then the A beta fibers will cover or interaction with S100A9 cores, and then to induce the AD.
Okay. So perhaps in the future, by blocking or inhibiting this from forming, then you may inhibit or slow down the progression AD.
Yeah. hopefully.
Okay, all right. Thank you.
Okay, thank you.
Okay, thank you, Dr. Eugene. Dr. Tashfik, do you have anything to add?
Okay, I think I have a last question for Gentao
Okay.
Pan. Okay,
Yeah.
all right. Can you go back to your replicate? Your MD simulation you run for 3 times, right? 100 nanosecond 3 times for both or 2 of the peptides, okay, and then 1 of the oli A.
Yeah.
Okay, all right. So can give me any reason why only 100 nanosecond and why you perform 3 times, three replicate. Why 100 nanosecond and 3 replicate in this your study?
Yeah, usually researchers will choose the 100 nanoseconds MD simulation, and then there are other professor have a lecture in USM before, and he mentioned that if we only do once MD simulation, we maybe miss some introduction. So for that we need to do more time. Such as 3 times MD simulation, or we have the longer MD simulation, such as the 500 or something else.
Okay, all right.
Yeah, to include all of the information.
Okay, all right. And then, so you mentioned that this oli A actually is a compound, right? It's not the peptide; it's a compound. Oli A.
Yeah. It's a natural product
It's a
from
natural product.
olive oil.
Okay, all right. And then, the significant interaction is pi-pi stacking, and also the hydrogen bond at residue. I not remember. Okay, this pi-pi stacking between tryptophan 88, okay, and hydrogen bond arginine 85. Okay, because that's why I said before, is it possible for you to analyze the hydrophobic interaction? Because only this compound, although it have its own structure and everything, but it can show the significance of the hydrophobic interaction between oli A and also the protein. Okay, so in your study, I think you have the trajectory. Okay, you have the trajectory 100 nanosecond, and then 3 replicate and everything. I think why not you just use like PLIP profile ligand interaction, protein ligand interaction profile, which is online and also can be run by command in your terminal, and then you analyze the interaction lah. That's my suggestion lah from me. Okay, in order for us to know either between the tryptophan 88 in the protein with your peptide can also establish this type of interaction, hydrophobic interaction. Then it can be considered good also as well as oli A. That's my suggestion, Jantau. Okay, you understand?
Okay.
Yeah, you can use, maybe you can use PLIP, protein ligand interaction profiler. That can online and also can use by the command from terminal. Then you can get all the interaction that involved in hydrophobic, in the hydrogen bond, okay, rather than you using the Amber lah, okay, that you can get from the MMGBSA Amber Python script. Okay, that's my suggestion lah. All right.
Okay.
And then I think that's all, Prof, from me. I think others is okay, no problem.
Thank you, Dr. Tashwik.
Okay, no problem. Okay.
Dr. Eugene, anything to add? No more from me. Yeah, I think no, that's all. Okay, thank you. If that's all, thank you, Jintao. And I would like to invite Dr. Chung to chair the next session. Thank you.
Okay, thank you, Dr. Ong. Our next student, just hold on. Our next student will be Song Jia-yi, supervised by Dr. Ong, and our evaluator will be Associate Professor Dr. Koo Boon Yin and Dr. Lang It Sin. So since Dr. Koo, you are there already, right?
Yes, yes.
Yeah, and Dr. Eric also, yes, that's still there, right?
Yes, yes.
Yeah, okay. In that case, Song Jia-yi, you can start your presentation now.
Can I see
thank
the
you, Doctor.
student for a while? Make sure
Sorry?
not AI is presenting.
Oh,
turn on the camera.
oh yeah.
Please turn on your video for a while.
Okay, okay.
Then after that you can after that you can start your presentations. Yes. Yeah. You can turn off the video.
Just hold on first, Song Jia-yi. It seems like your voice is on and off. Don't really can hear a clear sentence from you. The line seems like broken and on, like I hear broken. No, it's still the same.
Is that because of the video? It just because of the video, then maybe you can turn off.
Can you hear clearer now?
Yeah, it's much better. You can start now.
Okay. My research project is the illustration of mechanism of antiproliferative properties of Camelina communis against the cancer rhizolysis. And the introduction, Camelina communis is an annual plant belonging to the Camelina family, and native to East and Southeast Asia. And it has many ingredients such as flavonoids, alkaloids, and phyllosaccharides. And it also has anti-inflammatory, antibacterial, and antioxidant and antiviral effects. Literature review. Other plants on the Camelinaceae family and their extract have already shown a certain antiproliferative effect on certain cancer cells. And the Camelina communis only shown have the anti-inflammatory, antibacterial, and antioxidant effects. Based on this project's objective is to evaluate the antiproliferative effects on C. communis and explore its mechanism of regulating proliferation in cancer of rhizolysis and non-cancer rhizolysis. And its specific objective is to prepare aqueous extract of C. communis and assess antiproliferative properties of C. communis aqueous extract against cancer rhizolysis using MTT assay, and identify cell death pathway induced by C. communis aqueous extract against the cancer rhizolysis. The research hypothesis is the aqueous extract of C. communis can inhibit the proliferation of cancer rhizolysis, and the aqueous extract of C. communis is not toxic to non-cancer rhizolysis. The antiproliferative effect of the aqueous extract of C. communis may be achieved through inducing apoptosis or cell cycle arrest in cancer rhizolysis. The methodology is extract the plant into powder and use water boiling to do the extraction. And the optimal extract will do the sterilized test and then do the MTT with cytotoxicity assay. The drug concentration will 2 times zero dilution from 0.75 to 200 microgram per ml, and will detect after 24, 48, and 72 hours. And because the previous study has shown that 660 nanometer LED red line have a certain antiproliferative effect to the MCF7 and the MDMB231. This time we also detect after 24 hours plus LED 660 nanometer. Then we'll detect its cell death pathway, use flow cytometry to do cell cycle and apoptosis. This is this times after 24, 48, and 72 hours MTT results. First in MDMB231. At low concentration, the loss availability is 72 hours, but with the increased concentration, 24 hours got a loss availability. And
72 and the 24 survivability shows a decreased trade with increased concentration, but 48 hours is slightly increased. This may due to the different dose cycle. And in MCF7, this serialized at the concentration also shows the decreased trade, but at low concentration, 48 hours survivability is slightly higher than control. This may due to different cycle and the most times cytotoxic accumulated toxic. And in MCF7, 72 hours got a lost availability. And 48 hours is higher than the other two time points. In MCF10A, the changes is relatively small. And in 24 hours got a lost availability. The toxic is higher than 24 and 72 hours. At low concentration, it shows a decreased trade, but with increased concentration, 48, 24 and 72 shows a decreased trade, but 24 hours slightly increased. This may due to the high concentration got a highest accumulated toxin. And this crystallized in 24 hours. 24 hours MB231 got a lowest availability. And at the low concentration, MCF10A's toxic is very similar to MDMB231, but with a higher concentration. At the same concentration, Q reduce a certain amount of MDMB231. This actually also can kill a smaller amount of MCF10A and shows a low toxic to MCF10A when reduce a certain amount of 231. And the overall availability of MCF7 is slightly higher than MCF10A at a certain concentration. In 24 hours, this actually shows a higher toxic to MCF10A. And both MCF7 and the MDMB231's availability is higher than the MCF10A. So, and cause MCF10A is a normal cells. This is 48 hours this time point may not meaningful. And in 24 hours at a low concentration, this three cell lines survivability shows a decreased trade, and it's very similar. But at high concentration, MCF10A's availability is higher than MCF7 and MDMB231, shows a certain lower toxic to 10A, while have a higher toxic to both MCF7 and MDMB231. This last result is after 24 hours of treatment with extract LED 680 nanometer irradiation. First in MCF7, this realized all showed a decreased trade with increased concentration, but at low concentration, 50 LED is at the bottom. And with a higher concentration, the LED becomes the bottom. And cause this relay is very similar. LED here seems no additional effect on MCF7. In MDMB231, there shows a very low toxic to this 3 time point. And the 50 LED's availability is slightly higher than other 2 time point. In MCF10A, there are LED at the bottom, and 50 shows a certain low toxic to MCF10A, and 50 LED is slightly than 30 LED and 0 LED. That means this LED might interact with the drug and active some repair mechanism. That means we use extract to against the cancer cells. Short times LED may can reduce its toxin in normal cells, MSAP-10A. And in the already 24 hours, this result lines, this actually shows a good antiproliferative effect on MSAP-7 and shows a very low toxic to MDMB-231. And at the same concentration, reduce a certain amount of MSAP, certain amount of MSAP-7's availability will kill a very smaller, will reduce a very smaller amount of MSAP-10A's availability. In conclusion, this actually can reduce the availability of cancer cells with minimal impact on non-cancer cells. And the LED explorer may help mitigate the actual toxic effect on non-cancer cells. That means when use this extract to against the cancer ovarian cells, use LED explorer can reduce its adverse effect on normal cells. About the cell death pathway, this extract can increase the reactive oxygen species level to active some signaling pathways such as PI, such as inhibit PI3K, AKT, and the NF-kappa B signaling pathway to trigger the inflammatory response and induce some apoptosis. And in LED, use 660 nanometer LED radiation may can reduce its oxidative stress and inflammatory response in normal cells by regulating cell cycle factors P21 and P27, or active signaling pathway PI3K, AKT, and NF-kappa B to reduce a certain toxicity of this extract. This is my Gantt chart. I already finished the plant collection, identification, extraction, and the cytotoxicity assay. This month we'll do HPLC, cell cycle, and apoptosis detection. That's all. Thank you.
Okay, thank you, Jayu, for your presentation. So we shall now proceed to Q&A session. Any comments or questions from Dr. Gu first?
Oh, it is really shocking.
Yeah.
Can I see your Gantt chart again?
Okay.
So when do you need to submit your thesis?
Also in November.
November. So what is the month now?
November.
So you still have?
I still have cell cycle and apoptosis need to detect.
Are you sure you can finish within this month?
Maybe because this week and I already also prepared the cell cycle and apoptosis. We'll do it together.
Because I don't know how to comment. See your discussions. Go back. Yeah, one by one. Okay. Are you doing ROS?
No, I didn't do it.
Yeah, so you shouldn't. Are you going to detect any pathway?
Want to detect apoptosis.
Yeah, apoptosis is not. You're not going to detect anything on the pathway, right?
Yes.
Yeah, I think you may need to revise the discussions.
Okay.
Because your results are on proliferations, viability, right? MTT.
Yes, yes.
Exposed to your extract and also LED. Nothing on the pathway, nothing on the apoptosis, nothing on the ROS. Right.
Yes.
And then see your results again.
Okay. This is with LED.
Yeah. Can I see the results on the extract? Extract.
This is only extract.
Extracts. You are using crude extracts, right?
Yes.
Yes. Because your hypothesis, you want to show the toxicity, whether your extract toxic to the sunlight. Am I right?
Yes.
For your second. So this wasn't shown on your results because the MCF7 show reductions on the cell viability to about 60%. Yes. So you may discuss this. You are supposed to discuss your results. Okay.
Okay.
And I think another comments I mentioned it to you before. Your compounds display the potential cell viability effect until 24 hours only. Then after that, the effect drop, right?
Yes.
So from my point of view, you may consider my suggestions. You can adjust your study time point to 24 hours only. Perhaps you may adjust it to like 12 hours, 24 hours, 36 hours, and you do not need to expand the treatment time point until 72 hours unless you want to see what happened to the cells when they are resistant to the treatment. So, but you want to stick on that, it's okay, right? Let me see the results on the LED.
Please.
Okay. I agree with you. Exposed to the LED light reduce the toxicity, right? Just now you mentioned that, right?
Yes. Can reduce
But
the toxicity.
it's the line indicate on MCF7.
Yes.
The linear descending line.
LED only can reduce MCF7 stability.
I think the effect is not ideal because this is not the ideal. But this is not those. I'm not sure, but it looks like the effect is not ideal. Okay.
Yes.
Okay. Right. Maybe can I go back to your title? If this is you want to stick on the LED light study, you may need to revise your objective, your hypothesis, etc.
Okay.
When you're submitting your thesis. So elucidations on mechanisms of anti-proliferative. What mechanisms you are going to study?
It's so that pathways such as apoptosis and the cell cycle arrest.
So if you are not able to finish everything, maybe you may need to revise also the title, right?
Okay.
Stick on the elucidations on anti-proliferative property. I think this should be enough for your master study.
Okay.
Okay, thank you.
Thank you, doctor.
Okay, Song Jia, can you go back to your objective?
Objective, okay.
So you have 3 objectives. So far, what stage are you finished everything, all the 3 objectives?
No, I only finished the MTT. No, so that's
so 1, 2 only lah. 1 and 2.
Yes, 1, 2.
So the third one, can you finish? No,
Yeah, yes, can finish.
can finish, huh? Okay.
Yes.
At least three is good for mixed mode students. So can you go back to the MTT essay?
Okay.
So you compare, which one is your result? Which one is among all the result you want to show to us? You mean the most
The most
founding, which one is the best result?
is 24 hours.
24 hours. Can you explain some more?
At high concentration, it has a low toxic to amceptine, but have a higher toxic to 231, can kill a certain amount of 231 solubility.
So this is MTT assay. I think the variety is about 100. Okay, that is the best. So is which shell line is the best? I think the MCF7. MCF7
In this
MD
certain point is 231.
31.
Yes.
That's the best result you can get. The most sensitive in the MTT assay is most sensitive on the which cell line?
MDNB 231 in 24 hours.
Any reason for this explanation?
Okay, this difference may due to the different cell cycle and the different signaling pathway. In MDA and B231, this extract may inhibit its PI, 3K, and AKT signaling pathway, induce a cell death, and it has a slightly toxic to receptor A.
Okay, next slide. Can you go back? Next slide.
Okay, next please.
Next slide. This is the after treatment, no effect on the cell line. Okay, there's significant progression on CSIC50. So do you have any control here?
Control, yes, zero.
What?
There is no treatment and no LED.
Or no treatment? You mean the negative control? I mean the no treatment at all.
Yes.
Do you have other control? I mean the positive control for any compound? No.
No.
No, okay. Okay, next slide is last 9.
This is a conclusion.
Conclusion already. So I don't know. Like I seem like Dr. Koo also a bit short also. Our sudden we finish already. So maybe everything in a bit compact already because I can see many graph or chart in one slide already. So I hope you can finish all the third objective. Then final, have you start writing your thesis already?
Yes, I start writing introduction and methodology.
Oh, okay, good. Listen, good. I think you have one more month before you can complete the whole things. So you can complete the third one, huh? Confident, huh?
Okay.
The third objective. Sure. Okay, I think I have no questions as well, but I think first I think you need to highlight back during conclusion. I think you need to check back to your objective. So make sure you answer all the question. If not, you cannot achieve the objective, maybe you want to delete or discuss with your supervisor. That's my concern.
Okay, thank you, Doctor.
You cannot achieve, then probably you discuss back and maybe delete it or the other things or to revise back the objective. If not, look back not good on your thesis writing also and for your Viva presentations. I think that's all for me. Good luck. So,
okay, thank you, Doctor.
Okay.
Thank you, Dr. Rai. Is there any more last comments from Dr. Koo?
I think no more. No more.
Okay. If there is no
Maybe
more
your
further
comments on the
questions.
thesis later.
Yeah, during the Viva session. Okay, so thank you, Dr. Koo and Dr. Lai as the evaluator for Sung Jia Yi. So the morning session is done already. So we shall continue at 2 p.m. for another 3 students. The session will be chaired by Dr. Sasidaran. So with that, all the best and over and out. Bye-bye.
Thank you, Doctor.
Um,
Oh, okay.
After you can switch off.
Okay.
To ensure not AI is presenting.
Oh, okay.
All right. Okay. Okay.
Okay. Good afternoon, everyone. I'm Sultana B, and my supervisor is Dr. Ong Ming Tong. So previously, my project was a little bit different, but now there's a bit changes in the project. So now my title is Study on the Combined Cell Viability Effects of Lavender Essential Oil and 660 nanometer red light on MCF7 breast cancer cells. So previously I was focusing only on the effects of lavender essential oil, but now I'm combining the LED and the lavender essential oil to find the cell viability. So next is introduction. So as we all know, breast cancer is the most diagnosed cancer among women. It's the second most common cause of cancer-related deaths in women. Next, a little bit about lavender essential oil. It has been used for medicine in multiple countries for centuries because of its anxiolytic, anti-inflammatory, antioxidant, antimicrobial activities. Now, a little bit about the LED. So LED is a light-emitting diode. Specifically 660 and nanometer emits a red light. This is the picture of the LED, and this specific wavelength that I used in this is because it has various applications, particularly in horticulture, medical therapies, and scientific research. So my research hypothesis now is I'm gonna find the effects of both lavender essential oil and LED. Will they both combine will exhibit a significant anti-proliferatory effect on MCF7, which will be measured by MTT as IC50, indicating its significance as a therapeutic agent in breast cancer therapy. So my objectives now are to evaluate the cell viability of MCF7 using lavender essential oil, to evaluate cell viability of MCF7 against combined lavender essential oil and light emitting diode LED. Next is to evaluate cell proliferation using flow cytometry. So coming to literature review, first study demonstrated that linalool, which is present in lavender essential oil, has shown anti-proliferatory properties in various cancer cell lines. A second study has demonstrated that the ethanol extract of lavender has shown more cytotoxicity and safety profile against different breast cancer cell lines. Also, the in vivo study that they conducted showed a 43.29 reduction in tumor size with the ethanol extract. Another study demonstrated that the exposure to 660 nm LED light resulted in a 40% reduction in cell viability of breast cancer cell lines, that is MCF7 and MDA-MB231, just after 24 hours. So my research methodology is that I used Vanzo lavender essential oil and exposed it to LED for 15 minutes and 30 minutes and determined the anti-proliferatory effect. So for anti-proliferatory activity, this is the process that I followed. I first seeded the cells in a 96 well plate, then treated the cells after 24 hours with the lavender essential oil and incubated for 1 hour, and then treated with LED for 15 minutes and 30 minutes and incubated for 24 hours. After that, I went ahead with the MTD. So after adding MTD, I incubated for 4 hours. Then I removed everything and added 100 microliters of acidified isopropanol. And to dissolve the Parmesan crystals, next I added 100 microliter of distilled water to enhance the color and red absorbance as 570 nanometers. And then I plotted a graph for cell viability against the concentration. So as my sample is a lavender essential oil, the solubility is in it would require a solvent to dissolve the oil first. So I went with ethanol, but to determine if the ethanol will have effect on the cell, will it kill the cell more or will it make the cell proliferate more, at what concentration? I first did this experiment to determine the ethanol concentration that can be used to dissolve the lavender. So here we can see the blue line is for 24 hours, the green is 48, and the gray is 72. We can see that 72 hours, of course, it increases the cell viability a lot, and green is also significant. With 24 hours is still not that significant, but at after 1, there is a steep decline in the cell viability. So I have used 1.2% ethanol concentration, volume per volume, as my solvent to dissolve the lavender essential oil. So next, I treated the cells with MCF7 cells with lavender essential oil. So the highest concentration that I took was 2, and then I diluted it to 0.0125 till 0.125. So here we can see the cell viability has steeply decreased from 0 concentration to 0.125, and then the absorbance is still there. But when I saw when I observed the wells in the 96 under the microscope, I could not find cells that were giving off this absorbance. So this absorbance could be because of the oil itself, because I really couldn't see the cells in the wells. So yeah, next I treated the cells with lavender essential oil and with exposing the cells to LED for 15 minutes. Here we can see the cell viability has again steeply decreased for the concentration 0.125% volume per volume, and then the absorbance is slightly there. It's not significant, but it's there. But I couldn't see any cells in the well to show this absorbance. Yeah. Next, and again, I exposed the, there's a separate group that I exposed for 30 minutes. Here we can see the cell viability has again steeply decreased at 0.125% concentration, volume per volume. And it follows the same trend as before. Next, this is the combined result of the cell viability of all the 3 groups. The green is just the lavender essential oil. The yellow one is exposed for 15 minutes plus the lavender essential oil, and the red one is the lavender essential oil plus 30 minutes LED exposure. Here we can see the 0.125% volume per volume is steeply decreased from the 0 concentration, and the rest is not that significant because the error bars, they kind of overlap. So it's not a significant difference between the 3 groups. Yeah. So next I wanted to plot the IC50 for this, but there is no concentration to, there is no 50% decrease in the viability to plot for IC50. So I was not getting IC50 for this concentration, 0.125. So I decided to further dilute the concentration. So here I have diluted the concentrations of lavender essential oil from 0.125 to 0.0625 and 0.03125 and 0.15625. So now here we can see the cell viability has decreased 70% with the concentration 0.015625 and to 20% with 0.03125, and again the 0% of approximately 0% cell viability at 0.0625% of lavender essential oil. So and the absorbance, the rest of the absorbance could be an explanation of the lavender essential oil showing the absorbance itself, because I couldn't find the cells in the wells. So yeah, that could be a possible explanation for this absorbance. Next, this is the same as the previous graph. It's just that I used Prism graph pad to plot the IC50. So the IC50 of lavender essential oil on MCF7 is 0.0189% volume per volume. Next, so a summary of the results. So far is that I found that ethanol is safe till 1.2% volume per volume concentration, and lavender essential oil itself shows absorbance. That would be an explanation for the absorbance that we see at 2% concentration. Next, when treated with LED, the difference between the two groups of 15 minutes and the 30 minutes are not that significant because of the overlapping error bars. Next, the lavender essential oil is cytotoxic at concentration as low as 0.0625, and the IC50 of lavender essential oil was found to be 0.0189. Next, I also have work that is to be done in this month. So I have to replicate the experiment. This experiment, I have to replicate it because I did not go ahead with the LED for this one. I recently did this one to get the IC50. So I have to replicate this one with LED with the lowest lavender essential oil concentration. Next, I have to identify the composition of the lavender essential oil, and I also have to plan for flow cytometry. Next, the discussion. So according to a study, the decrease in cell viability by lavender essential oil is due to the increase in expression of caspase-3 gene. There is also a decrease in the expression of BCL2 gene, which is an anti-apoptotic. Next, because lavender majorly comprises of linalool, so this studies have found that this compound possesses significant anti-tumor properties, and the activity of linalool reflected that of the whole oil itself. Next, the study demonstrated that LED 660 NM promoted anti-proliferatory properties through autophagy. The next study demonstrated that lavender is cytotoxic to normal human mammary epithelial cells, but at a concentration of 0.25% volume per volume, which is higher than what I found in the MCF7 cells. Next, scope for future studies. So, because this linalool has potential anti tumor properties. So computer-aided drug designing can be used to modify the structure of this compound further to provide a safer and more predictable therapeutic agent. And this approach will combine the benefits of lavender and the computational techniques to enhance drug discovery and design processes. This is my gunshot. So previously, I planned to complete it in August. I plan to complete the cell viability assay in August, but I would still need 2 weeks in November to complete this. Next, for flow cytometry, I would be doing it in the 3rd week of November. And statistical analysis is a progressive analysis. These are my references. Thank you.
Okay, thank you, Sultana, for your presentation. So you have two evaluators, Dr. Eric and Dr. Kubunin. So I start with Dr. Associate Professor Dr. Kubunin. Any question or any suggestion? Dr. Ku, can you hear me?
Yes. Yeah.
Okay.
Another
Any
story.
question?
Huh?
You have any question?
Sultana, can I see your Gantt chart again?
Yes, doctor.
Huh? Yes? Again? So you will spend only 1 week for the flow cytometry.
Yeah.
Are you sure you can finish within a week?
I would hope it would finish in 1 week. I would try my best.
And what are you going to detect using flow cytometer?
I'm gonna detect the different characteristics of the cells. with the IC50 value of the lavender essential oil and also combining with the LED, I will find the different characteristics of this affected cells.
Different characteristic.
Like the cell size and the composition of the cells.
The the one microscopy study should do, right? But you can discuss with your supervisor. Can I see the discussion again? Before that, you have a slide on scope. Scope.
Yeah.
Scope, right? Scope for future study. The future study. I think this one also need to revise, huh? To enhance drug discovery. These are all very big therapeutic agents. Anyway, discuss with your supervisor. I don't think these are the future study. Future study at this moment. At this moment. Okay. See your discussions. Are you doing caspase 3 expression, BCL2 gene expressions? Are these your results?
Oh, no. This is from other studies, what they have found. So I still need this 2 weeks of time to conclude the expressions and the effect of LED, a definitive effect of LED on the cells.
So are these your hypothesis? You hypothesize the reduced proliferation is because of the increased expressions of caspase 3 and BCL2 gene expressions reductions. Because these are not from your study, maybe you should cite the reference first. Okay.
Okay, doctor.
I'm actually proud to commence your results. Maybe can you show me the last results, slide results? Last slide for your results. That one fluctuated a bit, fluctuated one.
You mean this one?
I think after you reduce the concentrations of LEO.
Yeah, this one.
This one?
Yeah.
How do you explain when the cell viability revive when you treat the cells with 2%, right, of LEO?
Yeah, when I when I got the reading, I wasn't sure why I was getting this, because in even in the previous one I got this slight absorbance. So when I did this experiment, so I was focusing on visually seeing the cells under the microscope. So at 0.03125, I still saw the cells at this absorbance, but I couldn't see the cells for this absorbance at 2. So the most likely explanation would be that it's a lavender essential oil that is showing this absorbance.
Did you state any anyway the number of cells you played for your essay?
I did not mention it, but for this experiment, I used 5,000 cells. For previous ones, I used 2,500 cells.
For 96 well plate, right?
Yeah.
5,000 cells. Yeah, I don't have, I can't comment. now. Maybe you just tell us what is the rationales to study essential oils, the effect of cell viability reductions of essential oils in combinations with LED. Why do you combine these two parameters for your study? No parameter.
Yeah, to answer that, I was thinking to see a more reduction in the cell viability when combined with LED. Like it would increase the, not increase, maybe it would decrease the cell viability when if it's combined with LED.
Okay, okay. I
Okay,
think
doctor.
thank you, Sultana.
Thank you,
Okay,
doctor.
thank you. Dr. Eric, any question or suggestion?
Okay, Sultana, can you go back to your title first?
Yes, doctor.
Combination effect of this. So your result is a combination for both of these method. One is the essential oil and one is the 660 nanometer wavelength of red light. Both.
Yeah.
Combination, right?
Yeah.
Okay. Understand. So what the reason, what the valid or problem statement or hypothesis that you have? You have a hypothesis here?
Yeah.
Okay, can you show me hypothesis? So yeah. So the lavender essential oil itself, it won't have a how? Which one? Is the LED gonna show the activity first, or essential oil gonna show the activity? After that, adding each other, they will increase the effect on the experiment. Which one? How the structure? First, you must have some basic to it. Which one start first? You mean the lavender oil or the red light?
So I'm gonna I mean
Understand my questions?
yeah.
Yeah,
So
okay.
I first added the lavender essential oil to the cells. I incubated for 1 hour and then I treated them with the LED for 30 minutes and 15 minutes. Then I incubated for 24 hours and got the absorbance.
So what the difference? What you expected? What you expected from your result from this hypothesis?
That it would decrease the cell viability. So
Do you find decrease? Do you find in the experiment your result?
yeah, that's what I'll explain in a bit. So here we can see that the LED effect of LED and the sample is not there is no much significant difference, but because I didn't had that these concentrations like further diluted, I had till only 0.125. I couldn't conclude the effect of LED because it's already reduced because of the lavender essential oil. So when I recently did this experiment where I diluted the concentration further, I found that now I could definitive have a result for the LED on the cells because the lavender essential oil is showing 70% of cell viability at this concentration. So if I add LED to this, I might conclude the LED result.
So you mean the LED give a more effect on compared to lavender? Based on the next slide.
It doesn't.
15.
I mean, here we can.
This one,
I
yes.
can. Yeah, here the difference is not significant to conclude it. So I still need to perform the LED on this.
Okay.
Yeah, on this concentration.
Sultana, I
Yeah.
have one suggestion actually, especially like the result is sometimes we cannot follow what the conclusion of it. Maybe after you presented one slide, maybe at the bottom you have one line or two line, the conclusion or anything, the result or the significance of the result that you want to highlight to people, so people will know. So you like this type, or maybe, and usually for cell viability, they usually use a line graph, so they can see the IC50. So I think it's, yeah, bar graph also okay, but usually people will use a line graph that you can see more for the IC50. I think this one you can discuss with supervisor. So my suggestion, you add one line, not say conclusion lah, maybe you have to highlight the significant, because sometimes when you, yes, you explain it, because I understand you explain the result, but sometimes we hardly can follow also, because sometimes online also this kind of thing hardly can follow. If you can put one line or two line on it, the result, or you want to highlight the significant of the result of this slide, then people will can follow easily than usual. Okay?
Yeah, okay, doctor.
Understand, eh?
Yeah.
Okay, I think you will do a statistical study also. This is triplicate, right? The result.
This is not triplicate.
Triplicate, okay. If you have one, the high, like you mentioned also, you only left about the flow cytometry experiment haven't done yet, right?
I still have to
How many experiments you not finished yet?
yeah, I still have to do the last graph experiment. I have to duplicate the experiment, but with LED, and I
Okay.
have to identify the composition and also cytometry.
How are you gonna identify the composition of LED, LEO?
Through HPLC.
Two?
HPLC.
SPLC.
Yeah.
Are you very familiar with SPLC, or you gonna ask other people to do it for you? Are you gonna do it by yourself or what?
I mean, I'll ask the person. I mean, they won't allow me to do it, so I have to take the sample to the chemical school, and they would run it for me.
Or you ask for servicing, okay? But at least even after servicing, I hope you can understand the principle of SPLC at least. When people ask you, then you know to explain the result also.
Yeah.
And yeah, I think because the first one I'm not very concerned. The second and the third one are the identify the composition, and maybe I'm not sure the composition of LEO actually. Probably you can search from literature review also. Maybe have some other study have done such kind of similar study. Maybe you can do a comparison, compare your result with the other people result. So find any difference. And I think the flow cytometry a bit rush also lah. Not sure lah. How many times you're gonna repeat the experiment also. I think I share some concern with Dr. Koo also. Maybe try your best. I understand that you're not necessary you cannot finish it, then leave it out of it, because especially in the objective or this kind of thing, you don't mention it. After that, just leave it up, and I think right. Have you started writing your thesis already?
No, I'm still working on the experiment. I'll start.
I think you already should start, and you only have 1 month left.
Yeah.
December you have to. Early December you have to you submit your thesis already, so you have to should write maybe a first 3 chapter already, and I think try to finish as possible. You don't have time left anymore. Okay, don't concentrate. If you cannot finish, I think discuss with supervisor. Sometimes, especially the flow cytometry is not the easier experiment, I have to say that. You need a few time to repeat and go, because the flow cytometry, we are not available here. I think either you go to IPPT or you use a service in iFarm. I think depend. Okay, because depend on the service also. Either you ask other people to do it for you, or you're gonna do it yourself. But either this kind of thing, but you need to know the principle of same like SPLC also. So at least the examiner asks you, then you can answer the principle there. Sometimes they will ask the principle or this kind of things. So at least you can interpret your result and give a good answer. But this can discuss with your supervisor. I think Dr. Tong is very wise expert in this kind of thing. Okay, that's all for me. Thank you very much.
Thank you, doctor.
Okay, thank you. Thank you, Dr. Eric. I have two questions. For the identification of the phytochemical, you're going to use GCMS or LCMS?
That I haven't looked into yet. I'm still stuck with my experiment, so I did not focus in the composition things.
So theoretically, what are you going to use? Do you have any idea? Liquid chromatography or gas chromatography? Okay, never mind, you can discuss with your supervisor. My next question, how you standardize your radiation with LED? Do you standardize? I mean the distance from the sample, all that.
I mean, there was previous work that was done with LED, so I just followed that thing. I mean, yeah.
No, follow means do you determine the distance, the light from the sample, all that?
Yeah. It is 10 centimeters distance between the cells and the light.
Okay, my last question. Can you tell us what the possible mechanism of LED to induce cell cytotoxicity? Theoretically.
Yeah. So it is like known that LED would directly affect the DNA of the cell. So that's how it would undergo autophagy, by directly connecting with affecting the DNA of the cells.
So if a person sit under the LED, he also will be affected. The DNA will affect him.
That is
I just give you some idea to think when you discuss your discussion later. So with that, I like to thanks Associate Professor Dr. Koo and Dr. Eric for their valuable suggestion and question. So with that, thank you. So best of luck, Sultan.
Thank you,
So
Doctor.
we go for next presenter.
Hey, hello. Dr. Sasi. It's a bit noisy
Yes.
here because my neighbor upstairs punctually. It's okay. So now we pass to the following person, Tapa. Okay, the next person who is going to present will be Tapa Maga Pushpa, student supervised by Associate Professor Dr. Sasidaran Sri Wasan. Okay. Now let's check evaluators now. Dr. Chiwailan, I have seen her around. And then Dr. Shahida. Let's see if she's around.
Hello.
Yeah, Dr. Shahida is around as well. So, Pushpa, you may start now.
Hello, Doctor. Can you hear my voice?
Yes, yes,
And can you see my slides?
yes.
Okay, thank you, Doctor. Good afternoon. Can I start?
Sure, please.
Okay. Good afternoon, everyone. I'm Puspa and Tapa, and the topic of my presentation is evaluation of antimicrobial activity of Alcea longifolia derived carbon dots against pathogenic microbes, and my supervisor is Dr. Sasi. So these are the contents: introduction, literature, review, problem of statements, objectives, hypothesis, methodology, results, and conclusion. Introduction. Carbon dots, these are fluorescent carbon-based nanoparticles. They are quasi-spherical, which means they are not entirely spherical. They have little bit of ridges on their surfaces. They are zero-dimensional, which means they cannot be measured. They are like a point dot. They do not have length, breadth, or width, and their size is 10 nanometer or less. And then they are composed of sp2 and sp3 carbon atoms, and their surface is surrounded by different functional groups such as amine, hydroxyl, carboxyl, etc. Advantages or some of the properties of carbon dots. Carbon dots, they have these versatile properties which can be used in different applications, and some of these are biocompatibility, environmental friendly, photostability. They are cost effective, multifunctionality. That means like their functions can be modified. They have high surface area, which helps in water soluble. And then it is easy to synthesize. And one of its important property is that it has some antimicrobial properties, which we are gonna talk a little bit so because of that, we're gonna see how it kills the organisms. So mechanism of action of carbon dots, there are like two mentioned in the literatures. One is in the absence of photon. So in the absence of photons, the carbon dots, they interact with the bacterial cells via diffusion or electrostatic interactions. And when they interact, they isolate these organisms from the growing cells and then deprive them of the nutritions or like stop them from spreading, which leads to the disruption of their physical metabolism, and then this leads to the death. And then another one is they prevent the formation of biofilm, which is one of the important environment needed by the bacteria. So if there's no film formation, bacteria, they end up dying. And also when carbon dots, once they're already inside the bacterial cells, they bind to the DNA and RNA molecules, and then denatures them and ends up killing the bacteria, as they cannot synthesize the needed protein molecules. Another one is in the presence of light. So once carbon dots, they absorb light, the electrons from the ground state, they go to the excited state. And then when these electrons come down to the ground state, they release some energy and then also some heat. And then at the same time, some of the electrons, what they do is they, instead of coming down directly, they go to the triplet state. And once they go to the triplet state, and while coming down, they release some phosphorescence, and at the same time, they release some oxygen, native oxygens, and then also ROS. And these ROS molecules, they bind to the different molecules of the microorganisms, and then prevents their functions, and then help to kill the bacteria. This is just a diagram of what I just said. Another one is Polyalcia longifolia. Since I'm going to use this, I use this plant, so a little bit on this. So Polyalcia, it is a evergreen medicinal plant. It is also known as like Indian mass tree, weeping tree, or asuka tree. It is classified under the family Annonaceae, and then it is found in the tropical and subtropical regions. It is native to Malaysia, Sri Lanka, India, and Bhutan. The tree is usually tall, and its leaves they droop down. They fall down to the ground and give this tree a slender kind of look, narrow and slender kind of look. And then little bit on leaf, as I'll be using, I use the leaves. So the leaves are usually 12 to 30 centimeter long and 3 to 7 centimeter wide. It has like wavy margins, and then has glossy dark green color. So the different parts of this plant has more than 20 compounds, or like more than dozens of phytochemical compounds such as flavonoids, alkaloids, diterpenes, and tannins. And like the majority of is the alkaloids and diterpenes. Traditional use. So poly means many and althea means cure in the Greek terms. So traditionally, this plant was used to, treat many diseases such as fever, skin disease, diabetes, rheumatic fever, body pain, etc. Some of the therapeutic properties of this plant are antioxidant, hypoglycemia, they are anti-inflammatory, anti-ulcer, antimicrobial. Literature review. So carbon dots was discovered in 2004 incidentally by Zhu and her gang while they were purifying these single wall nanotubes by electrophoresis. The carbon dots is composed of sp2 and sp3 carbon dots. So since its discovery, many researchers they strategize different approaches to synthesize carbon dots, but however they have not been able to, and since then their quest was to develop the best approach to synthesize carbon dots, and in this quest they recently started using these natural resources for the synthesis, and this thing is called green synthesis. So this is very simple, low cost, safe, and eco-friendly, and also it eliminates the need for passivation, which is like doping. So this was actually needed for the other kind of carbon dots. So this because the plants or natural resources have many compounds, it eliminates this passivation thing. And then so far, different natural resources such as banana peel, juice, lemon, walnut, neem, leaves, etc. have been used to synthesize carbon dots. So why carbon dots as antimicrobial agents? Well, there are several reasons. One of them is size and surface area. These carbon dots are so tiny that they can easily interact with the microbial agents, and then leading to the efficient antimicrobial activity. Another one is biocompatibility. Since they are derived from natural resources, they would have less negative effects compared to the conventional antibiotics. Another one is resistance minimization. The carbon dots, their mechanism of action is physical, and they do this oxidative damages, unlike our conventional antibiotics. So this minimizes the chances of resistance. And another one is functionalization. So since carbon dots contains different functional groups on the surfaces, it can be modified to target specific organisms of specific areas. And another one is it enhances antimicrobial activity. So studies have shown, some of the studies, for example, this Hamid Band, they used carbon dots in combination with antibiotics, and then they so that it had enhanced antimicrobial activity and biofilm inhibition. Gaps in the literature, there are many, but the one that kind of connects to my research is standardization issues. Carbon dots from the natural resources, like different methods and conditions are used, and because of this, there are no standard approach to synthesize carbon dots. So the problem of statements, one is rise of antimicrobial resistance, AMR, also known as antimicrobial resistance. It increases the it's the use of antimicrobial antibiotics in our daily life is causing the bacteria to become less effective, and then which is leading to a global threat. And also the AMR, it is leading to longer stay in the hospitals, and then they cause this high, and then it also has increased the mortality rate. So WHO estimated that by 2050, if this resistance continues, then about 10 million people would die annually. So another one is limitation of conventional antibiotics, which is like over-reliance on antibiotics. This leads to resistance. And then also it also has negative side effects. And another thing is like discovering these new antibiotics, it is costly and it takes a lot of time. So there is no any new antibiotics which can be used at the moment, which can deal with the current situation of AMR. Hence, new and alternative, effective, and sustainable antimicrobial agents are urgently required. The primary objective of this research is to synthesize carbon dots using P. longifolia leaves for an in vitro antibacterial investigation. Specific objectives are to synthesize carbon dots from P. longifolia leaves and to characterize them, and then to assess the antibacterial activity of the carbon dots obtained from P. longifolia leaves. Hypothesis: the synthesis of carbon dots from P. longifolia will result in efficient and reproducible methods for producing antibacterial nanoparticles. Then its characterization will reveal distinct structural and chemical properties, which will be suitable for antibacterial applications, and will exhibit significant antibacterial activity against the targeted bacterial strain in its in vitro studies. So this is the overall flowchart of my methodology. First, the leaves will be collected. Some of them were sent to the USM herbarium for identification, and then the remaining were washed, dried, and then cut into small pieces, and then went under pyrolysis, which gave brown color, converted into powder, and then dissolved into water, sonicated, centrifuged, and then filtered to, and then dry freeze to get carbon dots. The carbon dots were then characterized and then assessed. Its antimicrobial activity was assessed. So methodology for the synthesis of carbon dots. First of all, the leaves of P. longifolia was collected from the compound of USM. Some of them are sent to herbarium, and then some are washed, allowed to dry for 2 to 3 weeks, and then cut into pieces and turned into powder forms. The powder form was then heated at 300 degrees Celsius for 2 hours in a furnace, which gave brown residues. Then the residues was grounded into a powder form using mortar, and then dissolved in the water, then sonicated, centrifuged, and then filtered. The filtrate was then sent for dry freezing. And then this is the obtained carbon dots. So after synthesizing carbon dots, it was characterized using different techniques. One of them is UV visible spectral analysis. It was done to determine the optical properties or absorbance of the synthesized carbon dots. 3 ml of 10 microgram per ml carbon dot solution was dissolved, was transferred to a cuvette, and then its reading was taken in the wavelength in the range of 200 nanometer to 850 nanometer. Then TEM was carried out to determine the morphology and size distribution of the synthesized carbon dots. Then zeta potential, it was carried out to analyze the stability of the carbon dots and also to determine the surface charges. FTIR was carried out to identify the different functional groups present on the surface of the carbon dots. And then third objective, which is to assess the antimicrobial activity of carbon dots. For this, first of all, the bacterial culture was prepared. For this, the required materials were autoclaved, and then the stock culture was thawed and then inoculated in the nutrient media and then incubated at 37 degrees Celsius for 24 hours. And then it was transferred to LV broth and then incubated at 37 degrees Celsius for 24 hours in a shaker incubator. To assess the antimicrobial activity of carbon dots, the cultured bacteria in LV broth was taken and its OD was adjusted to 0.1 at 600 nanometer, and then the carbon dots was of different was taken and diluted in the PBS with different concentrations. After this, the bacterial suspension and the PBS with carbon dots of different concentration was seeded in the 96 well plates and then incubated in a second incubator at 37 degrees Celsius for 24 hours. And then OD was taken. So another one is time killing assay. It is used to assess the effectiveness of carbon dots over time on bacterial cells. For this, bacterial suspension, which equals to 0.5 Mark-Farland standard, and then carbon dots, which had a minimum inhibitory concentration, was taken and then seeded in the nutrient broth, and then it was incubated at 37 degrees Celsius. And then at the predetermined time, such as 0, 4, 8, 16, 20, 24, 28, and 32 hours, 1 ml was taken from each of the tubes, and then its OD was measured at 600 nanometer. For this, the control group was bacterial culture without CDs under identical conditions. Now, the results for the synthesis of carbon dots from P. longifolia. The carbon dots were synthesized using the pyrolysis method. This is the appearance of carbon dots in room light, and this is the water in comparison to the water. It appears pale yellow in color. So when it was exposed to UV light at 365 nanometer, the color of the fluorescence of the carbon dot appears green in color. Characterization of the carbon dots for the UV-vis spectral analysis. The observance of the carbon dot showed a peak at 227 nanometer, which attributed to the pi-pi transition. This means that the synthesized carbon dot absorb energy, absorb some light, and then also can release some energy. Some of the studies had higher absorption wavelengths, and some had lower compared to mine. TEM. This was done to study the morphology and size distribution. This is an image of a transmission electronic microscope. The carbon dots, they appear this dark and somewhat spherical in shape. The darker region corresponds to a higher concentration of carbon dots. This is a histogram for the size distribution of carbon dots, synthesized carbon dots. The carbon dots, the size of carbon dots, they lie between 2 nanometer and 6 nanometer. A peak is observed at 3 nanometer, where over 30% of the carbon dots are concentrated. So the size of the carbon dots are quite similar to other studies, such as Hong et al., whose size is 3.4 nanometer as average size. So these sizes, it shows that these sizes are within the range of carbon dots from other studies. So this shows the feature of carbon dots. Zeta potential, it was done to determine the stability of carbon dots in soluble form. The zeta potential was found to be minus 28.4 MV, which is a good, which indicates high stability of carbon dots. The negative charge repulses each other, and as a result, it prevents carbon dots from aggregation. So having higher negative charges is a good, as per the literatures. Other studies also have similar kind of zeta potentials. For example, Pitt et al. They had a carbon dot whose zeta potential was minus 20.8 MV. Also, this negative charge of carbon dots, it helps in antimicrobial activity, as it can enhance interactions within the bacterial cells. FTIR, this was the FTIR spectrum of carbon dot. It shows different peaks at different wavelengths. So these peaks represent different functional groups that are present in the carbon dots. For example, 3739, this corresponds to hydroxyl group, and 32 and 50 also correspond to a hydroxyl group. So there are many functional groups, and these functional groups, they can be used to, as I mentioned earlier, to modify these carbon dots to make it suitable for our own use. And then another one is these carbon dots can also, these functional groups can also play the role in the antibacterial activity. Third objective, which is to assess antimicrobial activity of carbon dots. Antimicrobial activity of carbon dots, irradiated carbon dots against bacillus subtilis, was carried out using MSc assay. For MSc assay, a wide range of concentration of carbon dots ranging from 5,000 microgram per mil to 330 microgram per mil was taken. The experiment was carried out in a triplicate. The statistical analysis was conducted using one-way ANOVA followed by turkey host hog test, and the 5,000 microgram per mil has the lowest OD, and 313 has the highest OD. This suggests that this carbon dot, the activity of the antimicrobial activity of carbon dot is dose dependent. Another one is time kill kinetic of white light irradiated of synthesized carbon dots. This was done to assess the antimicrobial effectiveness of the carbon dots. A student test was conducted to examine time-dependent antimicrobial activity. The treated group, it maintained a stable OD throughout the experiment, whereas the control group, it had a stable increase, and then after 10 hours, it has a sharp increase, which meant like it had great proliferation. Conclusion, carbon dots were successfully synthesized from P. longifolia and characterized for their structural, optical, and functional properties. Notably, carbon dots exhibited distinct green photoluminescence. white light irradiated carbon dots demonstrated significant inhibition of bacterial growth. So the overall conclusion for this project is that carbon dots derived from P. longifolia is a promising candidate as an alternative to antibiotics as an antimicrobial agent. The future research should focus on exploring their effectiveness in a broad range of pathogens, optimize synthesis protocols for consistent properties, and further understanding the mechanism of their antimicrobial activity. Thank you.
Okay, thank you, Puspa. Now I would like to invite Dr. Chiu to give your comments or ask any questions. Dr. Chiu. Dr. Chiu, hello. Or Dr. Shaida, do you want to start?
Yeah, I'm here, Dr. Ong. Maybe I
Yeah,
can.
yeah, Dr. Shaida, please go ahead.
Yeah, thank
Thank
you. Right, thank you. So Dr. Chiu, you want to continue?
Can you hear me?
Yeah, I can hear you. It's okay. Maybe you have sound problem, Dr. Chiu.
Yeah, never mind. Dr. Chiu, you just hold on first. Let Dr. Shaida go ahead. All right. Dr. Shaida, please go ahead. Thank you.
Thank you, Tapal. Nice work and nice presentation. A few questions from me. When you extract, not when you synthesize the carbon dots from the sources, the leaves, did you do any standardization, or you do you have different batches of the leaf, or you collect in one shot big bulk of the leaf and do synthesize?
For this, I collected the leaves in
Can
a
you show the
bulk.
slide when you show the flow from the leaf? Yeah, this one. Okay. So when you do the collection, is it in the bulk source or?
Yeah, I just took some from the tree
So you
without any.
do one time
Yeah.
collection.
Yes.
And then for the subsequent analysis, did you do any standardization in terms of, when you synthesize CD, you didn't check the concentration, right? It is based on, you verify it by light, using
Yes.
white light, and then you do the analysis on antimicrobial testing and so on. But
Yes.
do you know how much you can get carbon dots from how much of leaf?
That I don't know, but I did do quantum yield. I haven't put the result here, but because it was very low compared to other literatures, so.
So when you do testing on antimicrobial growth, can you show the slide where you do when you add the CD into yeah, this one. Previous slide. Yeah, this one. Okay, when you add, this is to grow the culture. So
This
you
is
do
the
dilution. So here you add different volume of CD, is it?
for here, like I take same amount of bacterial culture suspension and then same amount of CD solution.
Oh, same one stock solution of CD but different
concentration.
concentration of bacterial culture.
CD different same, like the bacterial suspension is same in all the tubes, but the concentration of the carbon dot is different.
But you just do dilution, right? You didn't check the concentration, right?
Double dilution. I did. So I mean, I did. So my initial concentration would be 5,000 microgram per mil, and then dilution, and then end up in 313.
For the 5,000.
Sorry?
How do you measure the 5,000? You mentioned just now you start with 5,000. Is that concentration microgram per mil?
Yeah.
So it means that.
Yes.
Yeah, okay. So it means that you did measure the concentration of carbon dots.
Yes.
After you synthesize.
Yes.
Okay, how do you measure? It is like protein, we can just use Bradford Bio-Rad assay to measure protein concentration. So for carbon dots, how did you measure the concentration?
For I mean, to use in the antimicrobial assessment, I like weight the carbon dots like 1 gram, like 5 mg per meal, and then I just diluted it in a twofold.
Oh, okay. So weight per volume. Okay. Okay. Now, you're saying that this carbon dot can substitute the use of antibiotics to kill the pathogen, right? So, what's your idea on how we can apply this? Is it similar like in solution form or in, when we want to give to patient? What is your idea? How can we deliver this? In what format?
I mean, first of all, the concentration at which this CD carbon dot is working is very high compared to our conventional antibiotics. So at the moment, it's not ready to use, but in the future, if we have to, if we find a way to synthesize it in such a way that we can use it in such a small amount, then one of the way that we can use it is by maybe we can mix it with the gel in like put gel and then just put it there, and then we can, before applying it or after applying it, we can irradiate with the light.
Okay.
Yeah, that is the only thing I can think of right now at the moment.
Okay. All right. And then so now your work is you start from like synthesis the CD from the source, the leaf, and then you do. I think because I just noticed the antimicrobial growth when you do this specific objective 3. There's a part where you sorry. I think this is in specific objective 3, you do antimicrobial growth and MIC tests, right? Sorry, I wrongly think it different way. Okay, I think this is the last question from me. How do you irradiate CD?
In my case, I irradiate it with my white light. I just expose it to the light.
Is there any specific protocol for how long?
No, there isn't any specific protocol. There isn't any specific protocol for the synthesis of carbon dots. Like everyone is, just exploring. So this project or this research is also for the exploration. To see if we can find out any to see if we can find out a protocol. So it's not there isn't any standard way to do anything.
Everyone is still trying to establish protocol for CD.
Yeah.
All right. Yeah, I understand. Okay, I think that's all from me, Dr. Ong. Thank you for your answer. And
Yeah, thank you, Dr. Shaida. Thank you. Dr. Chiu, are you ready?
Can you hear me?
Yes.
Yeah.
Dr. Chiu, please go ahead.
Yeah, Tapa. First question is, you mentioned that the CD is zero dimension in your intro, and you claim that it has high surface area as its advantages. So, are these two contrasting themselves?
Yeah, it has a not really. When it means the high surface, it means
Yeah,
like it has.
in your advantages of this, you said it's easy to be synthesized and it has high surface area and all that, right? Yeah, you have all the advantages of CDs, right? The fourth, yeah, high surface area here, and you said zero dimension, so I'm a bit confused here. What do you mean by zero dimension? Then it has high surface area.
I'm not quite sure about this. I mean, high surface area, it means like it's so dense and it has so many functional groups. Like it can attach, like these functional groups can attach to it, to the bacterial cell wall. So it's in that sense.
Yeah, because I'm a bit doubtful about this zero dimensional claim, because you can measure the size and you can, like, right, you do you do mention the range of sizes, right? Then how can it be zero dimensional? You did characterize the CD, right? The size of the CD.
Yeah.
Yeah, to me, to my understanding, if it's zero dimensional, then how can you measure it?
I think that's possible because of the TM, because it's done in a very high magnification.
Yeah, anyhow, to me, if you want to measure a size of something, then it has to be a dimension, right? So that you can measure.
Yeah.
Yeah. Okay, maybe you find out more about that and clarify lah, maybe in your write up and all that. Okay, because to my understanding lah, to me zero dimensional is like, I don't know. So if to measure, then you need a dimension, right? Then for the phytochemicals, right? You claim that the longifolia, you have over 20 plus phytochemicals inside, right? So can you theoretically hypothesize, like you have like banana peel, wool, like other source, right? To synthesize carbon dots. So at the end of all this synthesis, is there any difference between the different natural sources, or it will boil down to one same carbon dots?
Yeah, it doesn't come out to the same carbon dots. This compound, these different compounds also influence the functional group of the carbon dots, and maybe not all natural resources would have the same kind of phytochemicals. So based on the natural resources used and the method used, it's not gonna give you so far it has not given any same carbon dots with the exact similar features. So I would say it's not possible at the moment.
Anyhow, it could be different, but anyhow, it will boils out to different functional groups. So, right? Can I say that it will give you different functional group combination of functional groups? Is it so?
Yes. according to the literatures, not so far I've read, not all the carbon dots that were synthesized from different sources had same features. Everybody had different. They were in that range, particular range, but not the exact. So it's not going to be same.
They will be different in terms of functional groups, right? Different functional groups.
Different functional groups, and then also
What else?
their observance, like the wavelength at which they observe lights and then emit.
Oh, okay. So can I go to your slide 22? Yeah, you can see that here, the stock, right, at the right hand side, the stock you thaw it and you inoculate directly. How old is the stock? Your culture stock?
It's not that old.
Meaning how roughly?
I mean,
My
I
question
think
is, can it grow? I mean, grow well if you thaw directly and inoculate directly onto the plate, the agar plate, without reviving again the stock
yeah,
culture.
this is to revive the culture.
Oh, meaning you revive the culture. There's a step
Yeah.
missing, right? You should revive the
Oh
stock
yeah,
instead of you thaw it and you inoculate, right? You
yes.
did
I did
revive
revive
it
it.
first.
Yeah, I did. Yeah, I did revive it first. I just missed the picture here.
Oh, okay. So slide 26. So here, right, you see the peak at 227. I just would like to know from you whether you have read somewhere else in your literature review. Is there other peak reported for other CDs, other carbon dots?
Yeah, yes. There are some. I mean, there are other peaks reported in the papers. One of them had a peak at 220 nanometer, which is low to mine. And then this is the one that I've found, which was close to mine. And then other papers, this peak was at much higher wavelength, around 270 or 280s, 267.
So it's all below 300. It's around 200 something.
So yeah, the absorption, according to the papers, this absorption ranges from between 200 to 300 nanometers.
Okay. So if you see here, you claim that this 227 is correlate associated with your carbon-carbon double bonds, right?
Mm-hmm.
Your conclusion. So this is slide 26. If you go to slide 30, here your FTIR show many peaks of different functional groups and or bonds, right? Right, CO, COC, CH. You can see quite a number of peaks here, right? Different bonds, I would say, right?
Mm-hmm.
So how come all these are not reflected in your slide 26? I mean your UV spectra. It didn't show all these other type of bondings, right? Or functional groups, I would say. It shows only the CC, right? How about other? Yeah, it's not captured here. You capture only CC, right? 227. So what happened to other peaks captured in your FTIR?
I am not for the UVs, I haven't really seen a paper where they have mentioned this the sole purpose of this FTIR is to see the functional groups, and the purpose of UVs is to see the absorbance. So I haven't seen any paper where they have, mentioned other bonds like in the FTIR. So I'm not sure how I'm supposed to do that.
Okay. Okay, then how about slide 29? Yeah, your I just would like to know what is the range of this stability, if is what is the range of this zeta potential distribution that shows stability, the item is stable within from what range to what range?
So for this, any carbon dots that shows negative charge, which is like not in between the 30 and positive 30 and negative 30. So
30.
positive 30, if there's any value in between positive 30 and negative 30, that is not considered stable. So any other value apart from that is considered a good, is considered good.
Stable. Okay. My last questions. Could you see any possible limitation or side effects of carbon dots
Possible side effects.
or limitation of these carbon dots?
So far, there's no standard protocol to synthesize it.
No, any possible side effects or limitation?
No, not really.
Okay. I think that's all from me. Thank you, Tapa. Dr. Ong.
Thank you, Dr.
Okay, thank you, Dr. Chiu. Dr. Shaida, do you have any further questions to ask?
No, Dr. Ong. No.
Okay, if that's the case, then thank you, Papa. We stop here for you. And then I would like to invite Dr. Sasi to chair the next session.
Thank you,
Thank
Doctor.
you. Dr. Sasi is not around. Yeah, Dr. Sasi is around. Yeah.
Yeah.
Yeah. Okay. Pass to you
I
now.
using handphone, that's why slow, little bit slow.
It's okay. It's okay.
My
So.
computer updating. So
Uh-huh.
thank you, Dr.
Yeah.
Wang. Thanks for
Okay.
chatting
Thank you,
for
Dr.
this part.
Sasi.
Thank you very much.
Okay.
So we go for next presenter.
Yeah.
Yeah. So the next presenter is Yap Zijian under the supervision of Dr. Eugene. So can you share the slide first?
Okay.
And the examiner for Yap Zijian is Dr. Fatma and Dr. Nick Hughes. Okay, Yap, can you share the slide?
All right. Wow, please bear with me. Are you all able to see the slide?
Yeah, it's uploading. Okay, now I can see. Okay, you can start your presentation.
Doktor ni tak ada lagi, Doktor.
Sorry?
Doktor ni just tak ada lagi dalam tunggu sebentar.
Wait, Dr. Nick not yet.
Sekejap ya, let me check. Let me check on her whether she's there or not.
So Leo, wait for a while.
Okay,
Waiting,
no
waiting
problem.
for another examiner. Okay. Dr. Nick boleh dengar?
Boleh, Dr. Sasi.
Okay, so sudah akan present sekarang.
Okay.
Okay, Leo can proceed, please.
All right. Very good afternoon. Okay, thank you for coming to my presentation show. Okay. Good afternoon to my supervisors, Dr. Eugene, co-supervisors, Dr. James Liu and Dr. Li Wei Tang from Codon Genomics and Berhad, and also my examiners, Dr. Fama and Dr. Nick, and my fellow friends. Okay, so today I'm going to present about my dissertation. Okay, my current progress on my dissertation, which topic is machine learning based prediction method in identifying skin cancer type based on or by microRNA expression. Okay, so to the next slide.
Ibu boleh tolong mi ambil tak?
Sorry. Okay, sure.
Proceed.
Okay, I just proceed.
Pak macam ni, almari macam ni, Min letak semua kat tu, bantu Pak letak semua. Dah.
Dr. Saida, off your mic. Thank you.
Tak, buku itu macam ni kan.
Can you all hear me now? Okay, good.
Okay. Proceed.
So just a brief or a quick intro about what is going on with my project. So skin cancer is characterized by abnormal growth of skin cells, mainly due to UV ray exposures, and causes a DNA mutation that trigger uncontrolled skin proliferation, leading to tumor formation. So generally, there's two types of cells. When it's uncontrollably proliferate, it will lead to skin cancer. So one is melanoma, which originates from your melanocytes, which is the pigment cells, okay, pigment producing cells. And also the non-melanoma cells, which originates from our epidermis cells, for example, squamous cell carcinoma and basal cell carcinoma. So a conventional diagnosis of skin cancer is basically based on the visual inspection on the biopsies, visual inspection and also inspection on the biopsies. And one of the downfall of this method is prone to subjectivity, time consuming, and could have some potential error that leads to some others misdiagnosis of the disease. Okay, good. Next. So the problem statement of my dissertation is traditional skin cancer diagnosis method are reliant on visual inspection, which may have a lot of disadvantage, like mentioned just now. So this project proposed using microRNA expression profile based on predictive models to improve diagnosis accuracy and efficiency by identifying specific microRNA profiles or expression profiles associated with their corresponding skin cancer types. So the objective of this dissertation, the general aim is to establish a machine learning pipeline in predicting skin cancer type based on microRNA expression profile. So what I mean by this objective is that I'm hoping to establish a pipeline that you can use or someone else also can use to make a prediction models, okay, to predict skin cancers. So the expected outcomes or generally what I want to achieve on here is that from this dissertation, a more specific one is able to identify a candidate microRNA data set that serve as a biomarkers for these skin cancer types, and able to develop and test predictive models, for diagnosis. So the general workflow of my dissertation is, of course, we'll start with data mining. Then after that, we'll do feature selections. And after feature selection, we'll do training of prediction models. And after that, we'll do validation. So what are these? I'll be going through a few on the next slides. So data mining, I just call this as phase objective of what I'm trying to achieve from here. So it's like what it says, data mining is to retrieve a microRNA expression profile data that accurately distinguish skin cancer types. This is pretty straightforward. Okay, for feature selection, so you might be wondering how we do feature selections. So I'll go through the method after this, but I'll tell you about the objective. The general aim of this is to identify and filter the most relevant microRNA features that contribute to accurate predicting skin cancer types. And so you can reduce the dimensionality and improve, sorry, typo here, the efficiency and accuracy of the predictive models. Okay, so what does it mean by this is okay, so as we know that there are many types of microRNA that's affecting, it may go to hundreds of thousands for particular phenotypes. So that applies on our situation here as well. So from the microRNA data, the reason why we have this is we want to filter out some of the relevant one. We can't have too many data to fit into prediction models. Yeah. So for example, in my case, at first I got 2,000 types of microRNA that's setting up as a parameter. So from there, I need to filter down and cut down into slim down so that I can improve the efficiency of my prediction models and also the accuracy. So the third one is, of course, prediction models training. After that, we got the selected or the microRNA features that is relevant. So we will put this to develop a machine learning models that accurately classify skin cancer types based on what is being selected from the previous phase. And the last one will be validation. Validations to evaluate the predictive model's accuracy, okay, and generalized ability in classifying skin cancer types. So basically is to validate how accurate is this thing, and also potential analysis on here. I'm still not sure about this phase what I'm going to do yet, but so far what I'm confirmed is I will do validation. That's of course. Now for literature review. So first, I would like to talk about overview of microRNA. MicroRNAs are small non-coding RNA that regulate gene expression by binding to mRNA. Okay, so microRNA act as a fine-tuners of protein synthesis and are involved in multiple cancer-related pathways. So microRNA in cancers. So there's impacts of microRNA in cancers. So of course they can act as oncogenic microRNAs that switch on some of the oncogenic genes. Some can act as tumor suppressor. Okay, and yeah, so yeah, involved in multiple cancer hallmarks, influencing the proliferation, apoptosis, and metastasis. So this is like the biogenesis of microRNA. This is how the microRNA is being produced and what it could be bring. So from the figure here, you can see microRNA is also is transcribed. Is also the template is located in our DNA. It's wrapped in our DNA. So after it's been translated, it will become a primary microRNA, and with some other enzyme, it will become Yeah, so the middle one is just a process of how it get matured. I'm not going to deep for this. Okay, and then after that, the matured one will bind with a protein, and these two things, this complex will bind with your microRNA that affects some of your biological process like mRNA target cleavage, translational repression, and mRNA degradation. So the more important thing is about machine learning. So allow me to do some small introduction on machine learning. So machine learning is widely applied in cancer research. It's very common nowadays, helping in early detection, diagnosis, and personalized treatment. So ML models or machine learning models analyze complex data sets and identifying patterns and predicting outcomes. So in skin cancer, what machine learning can help do is, or to help us to achieve is, we can improve our diagnostic accuracy by identifying key biomarkers. So aid in categorizing cancer types, like what I've been doing in my project. And also ML methods such as supervised learning enable models to learn from unlabeled data. Yeah, so this is basically this the same thing about from number 2. Okay, so in this presentation, I'm going to mainly talk about supervised learning because this is the method that we'll be using in our dissertation. The other two is not relevant in this dissertation as we already have the outcome and we already have the control variables and the responding variables, like the outcome and the variables. So for these other two that are not circled is actually you are trying to predict something that is without and responding variables. Yeah, you can understand in this way. So I'm going to talk about supervised. The method that we use in this dissertation is called supervised learning. So definition, machine learning is a method to train computers. So yeah, next slide. So supervised machine learning. So supervised machine learning involves training a model on label data. So a label data consists of responding variables and control variables, where both input and output are known. Yeah, that's what I'm saying. So the model learns the relationship between inputs and outputs to make prediction on new or unseen data. Okay, so this is some of the common algorithms that is in cancer prediction. This is very widely used. So SVM, decision tree, and random forest. So the advantage of using the supervised learning is ability to classify and predict outcomes, okay, from a label data. That's very important. Now, also allow me to introduce some of the database that I use. So there are three databases mainly. So there's Jaw Omnibus. This is part of the database that is from NCBI, if I'm not mistaken. So the gene expression omnibus is a public repository of high throughput gene expression data. So the second one is TCGA, it's called the Cancer Genome Atlas. Okay. And then the third one is microRNA base, is a database dedicated to microRNA sequence and notations. So okay. So next, what is feature selections? So yeah, so here I will do more details explanation about feature selection. So feature selection is the process of selecting a subset of relevant features from a large data set. Like I mentioned just now, my current situation, I have 2,000 of controlling variables and only 2 types of responding variables. So that's why I need to do feature selection. So because I need to process selecting a subset relevant features from large data sets. So it's a crucial step in machine learning and data analysis as it can help to improve model performance, reduce computational cost, and also enhance interpretability. So the advantage of doing feature selection is, of course, get to reduce noise, get to improve model performance, and enhance interpretability. So these are the description for that. I'm going to waste time on this. So about type of feature selections. So what are the type of feature selection we have? So generally there's 3. So there are filter methods, embedded methods, and shrinkage method. So filter method is more like what you have, like what we do with our experiment usually. Okay, so maybe you could use chi-square test to see some relevant features. Yeah, so these methods evaluate features individually based on their statistical properties. Yeah, and then the second one is embedded method. This method evaluate a subset of features by training and testing a model on each subset. So the example of this is called RFE, recursive feature elimination. This two is not important. So the third one is more or less important because this is going to be features in our dissertation, in my dissertation. So shrinkage method, this method incorporates feature selection as part of the model training process. So example of that is called, we have L1 and L2 regularization. Yeah, I think mispronounced. So shrinkage method. So shrinkage method is a subset selection method. Use least square to fit a linear model that contains of subset of predictors. So as an alternative, we can train a model containing all P predictors using a technique. Okay, so it may not be immediately obvious why such a constraint should improve the fit, but it turns out that shrinkage the coefficient estimate can significantly reduce their variance. Now, I know this text is very boring and also very confusing. That's why I make a simple slide on the next one to explain to you about this. Okay, so let's go to the basics. So what we have, so what we do usually for our experiment is that, of course, there's responding variables here. okay, and also there's controlling variables. So we will plot the data on the graph, and then we plot there, and then of course at the end of the data we will produce a best fit line to fit all the data points, right? So okay, so this is what we do usually. So that is so note that these arrows is what we call the coefficient, the R square that's being calculated. So after you do your experiment, probably you will see how accurate is your best fit line, right? So you will do this R square test. So this R square test is generated by this formula. Okay, this is in words and this is in mathematical terms. Okay, so the R square test in statistics studies is also sometimes called coefficient. So don't be confused when I say coefficient, it's actually referring to this R square that you usually do in your experiment. So let's go back to this our slide. So how can these regularization methods L1 and L2 can help in our objective? Okay, so regularization is a technique used to prevent overfitting by penalizing large coefficients. Okay, so as you can see by these formulas, right? So these formulas, right? Just now I told you that when you have data points, you'll try to do the best fit line to see this line whether is whether fit or not to fit as much as the data points I want, right? So what if I tell you actually there's another way of actually to play around with the data. So in today's objective, sorry, what I'm trying to do is need to narrow down the amounts of features, right? So these features I will be subbing. So these data points so these features will have data points, and these data points I will generate a number for that, and it will be subbing into these following formulas. So you can ignore this front part one. You can just focus on the last part one, this Bj square and lambda. So this Bj square is actually the coefficient, which is the R square that you do. Okay, so I can change the line by changing the number at this behind. I can change the best fit line by changing this number behind here, by manipulating the numbers at here. So why do I want to do that? Because I want to see what data is relevant, what features is relevant in our case studies. So that's why I will try to manipulate the line to do overfitting. So overfitting is actually means I will connect the point as much as possible and ignore some of the other points, and from there I produce a overfitting line from there. Yeah. So what's the difference between L1 and L2? So as you can see, there is a slight difference on this Bj and this Bj square. Okay, so L1 regularization adds penalty equals to the absolute value of the coefficient. So this we'll put a modular here, okay, to make this plus and minus, and carrying sum of coefficient to be 0. So as we know, we do R square usually when you do R square, when you square both values, right, you won't able to get 0, right? So this method is used in ridge regression, but in our lasso regularization or regression is a bit different, whereby we don't square them together. We make them as a 0. The reason why we want to make them as a 0, because we want to see, because the coefficient is referred to the distance of your data points to the line. So if we can make them as a 0, it means that this data is not relevant in our studies. So from there we can eliminate some of this data. Okay, so both methods help to improve model generalization, but L1 is especially effective in our cases. So you will see why later in our result. Okay, let's skip this. Okay, so method and result. So at first, please allow me also to talk about data mining. So this is the one I did not present on previous presentation. Apologies for this. So for data obtained from the geo and microRNA database, the data mining process was very straightforward, involving only the use of search key and filters. The query result were downloaded as a zip file, and which was then extracted into a normal file, and the data was in TXT format, but is later to convert to Excel format for easier reading. So this is the general workflow of how I get the data from Geo Omnibus. Okay, so you go to this website and then you type the keyword you want to search here. For example, I type basal cell carcinoma microRNA. Ignore this S, this is what typo as well. So from here, okay, you will click on these 15 numbers. Then when you click this, it will lead you to this page. So this page will show studies. Okay, this is the description and also the title of the studies. So from there you can see. Then after that, if there's too much of the result appears at here, you can also filter by customizing the study type. Of course, here I customize by searching for expression profiling by high throughput sequencing. Okay, I'm not using an array data, but I'm using a sequencing data. Okay, so yeah, so you can see the type non-coding RNA. Non-coding RNA refers to our microRNA here, and profiling by high throughput sequencing. So this is how I got my data from Geo Omnibus. For microRNA base, I didn't show here, but actually it's also straightforward. But the data was only one data that I tried to extract. So I just searched and yeah. The next one is TCGA. Data retrieved from TCGA begin by searching with specific keywords, also same with that, such as in our case, like maybe I want to search cutaneous melanoma to filter relevant entries. Then the selected data set will then added to the card and manifest file that record the ID of the project was generated. And why do I want to do this manifest file? Because I will use these GDC data transfer tools to read the manifest file and get the data I want. So this is the UI when you enter the GDC data repository. So from here you can see also you click on this repository, you'll get this page. And from there, I filter my sites, primary sites, and also disease type. Okay, then it will show you the result you got, and then also we can further filter by clicking some of these features. Okay, and get what you want. So after this, you can choose to add all these things into a cart. This is your cart, like shopping, you go to a cart. And now you go to the cart page, you download the manifest file. Okay, and this manifest file, you require to download the GDC data transfer tool and download. And by using it will read the manifest file and you will download the corresponding data from there. All right, so this is only the only very small part of my data. As you can see here, this is the sample microRNA. This is the study samples, and on top of that is the microRNA names types. Okay, some of the name nomenclatures are actually outdated. Some of them are not, but I managed to arrange them all together. So this is part of that also. Okay, so I'm going to talk about what data is being chosen to study to move on to the next phase, chapter 2, which is feature selection. So out of these data, only I get to manage to select 2 data from Geo Omnibus. The reason why I did that is because, so as you can see from here, so this is the data from one repository, and this is the microRNA. So as you can see that some of these microRNA sequence is not consistent because some of the study was carried out for some times ago from TCGA, and also the nomenclature is also outdated, like this one, HSA LED 7A2*1. No people is using this anymore. So that's why I rejected some of the data that has this nomenclature and do on this. So after that, I rejected. I only managed to get the 2 data, the ID that I used to proceed. Now, as you can see from here, just now I have a lot of microRNA name type, right? So you can see some of the data are not available NA. And some of the data, the microRNA is actually overlapping. Due to the nomenclature issue, is overlapping. So the microRNA parameter being sequenced is not consistent. So at the end of the day, I only get these two studies which has the consistent microRNA being tested on the sequencing platform. Okay, so finally I get these two lah. So one is representing the data for basal cell carcinoma, and the other one is for melanoma. Okay, chapter 2, feature selection. So before we do feature selection, we need to pre-process our data. So the data was inspected to ensure it has no missing values, i.e. NA, like just now the NA. Okay. The data point with NA also in the two ID just now, right? They also have data point with NA. So I also need to convert them into 0 for my analysis, for my downstream analysis. Okay, so after we inspect the data, we standardize the data by transforming each data point to a Z-score formula, Z-score by following formula. So this is the formula on how you transfer the data from the Z-score, where X is refers to the original data point. This is the original value that is in your data. U or mu, wait, is this mu? I guess. Oh, just call it mean. Okay, mean. Mean is the mean of the whole column of the data. What I mean by column is that this. So this whole mean of this, and then followed by the standard deviation. Okay. So after I transform it into a Z-score, the data set was partitioned into training and testing set using an 80 and 20% split. So 80 would be for the testing and 20 for the training. Okay. So the reason why we do the third step here is we want to cross-validate the feature selection, to cross-validate the features being selected from the feature selection. Yeah. Okay. So then the new data was incorporated into 3 different models to do feature selection or to test. So we have logistic regression. This is how a graph of logistic regression looks like. We will try to fit the data point as much as it has a curve to look like this. This is logistic regression. Then we have a ridge regression, which is a linear regression, but we play around with the best fit line, and also lasso, which is a brother of a ridge regression also. But just a slightly different on how we do the handling the coefficient. Okay, so for the result, for logistic regression was also not quite ideal in my study. Throughout my study, I found that it's not so ideal. The reason why is, okay, so from here, right, as you can see, I will show you the script later and how I do this. Okay, but from here you can see that I really tested with logistic models, and I try to extract the coefficient. That's why, as you can see, there's a coefficient here. So, and this coefficient will be converted into p-value for studies. Okay, so this p-value that is less than 0.05 will be used lah. But as you can see, when I try to print the significant features based on the p-value that I set here, it shows 0. It means that no features that have p-value that is less than 0.05, which means all the features are significantly different, which doesn't achieve our agenda. Okay, so same thing happens for ridge regression. So as you can see, after I do and I try to extract the significant ridge features, it shows that I still have 1,000 or 1,000, and actually it extend to 2,000 more of the data. So in other words, actually I can say that ridge regression did not do anything to my data set, so which is not contributing to our agenda also. So which leads to, so a discussion for this is that why does not work? Why this doesn't work is because due to high dimensionality and multi-collinearity. High dimensionality usually refers to I have too many features on there, and multi-collinearity is that our feature is highly correlating to each others. Okay, and also which this does not fit our purpose. So the reason, this is the two reasons why these two methods I will be not choosing to use, and I will do the third one, okay, which is lasso, okay. Okay, so this is the result for lasso regression. So this S1, please ignore because I did not rename this S1, but this S1 is actually refers to the coefficients. So as you can see that from this S1, there's a lot of, what is it appear here? A lot of zero. So zero, it means that this data is not significantly different from our. So this data is not significantly different from our. It's not significantly different. Hence, you can say that this data from these features are contributing to our studies. Okay, when it sets to 0. So these are the significant lasso features. Okay, remember these are coefficient. So this means that this data point is actually very far away from our overfitting line, and thus we can conclude that these features are significant because it's very far away, so that means that this has something to do with the expression file. The expression count has something to do with the outcome lah. Right. So also I made a regression for doing the I'm sorry, what am I talking? Sorry. Also I made a histogram to look at the feature selection frequency. Okay. So of course, from the lasso, how can I say these results are valid? How I validate this? Okay. So what I did is that I did bootstrap for 1,000 times, which means I repeat the feature selection process for 1,000 times, and this is after I repeat for 1,000 times, I make it into percentage to see how much of the features appear in more than a number. Yeah. So this line I draw is the threshold I use. So from this threshold line, I will choose the data, proceed to our next phase, which is prediction models. And these data are not considered on the next phase already. Why? Because as you can see, the selection frequency is actually very low. For 1,000 times, I repeat the process, right? So these are the data that appear quite a lot number of times. So that's why it has very high frequency, selection frequency. Okay, good. So the next also I draw a distribution or histogram distribution on the bootstrap accuracy. So as you can see that if the accuracy reach almost the max if I do the bootstrap for around 60 times. But of course I do 1,000 times. 1,000 I think is not showing here. Okay, so yeah. So yeah, so that's basically that ends our lasso about our feature selection, right? So this is the final microRNA data I'm going to use to build my models. So talk about a future progress. So this is the milestone I set for my own dissertation. So from here, I think we are on November right now. By now, I have to do data validation. But now currently, I'm still stuck on data modeling. Not stuck, but I'm on this stage. I already, I think there's 5 here, KNN, SVM. I don't know why it doesn't show. DT, RS, and XGBoost. These are the short form for the models. Okay, the name for the models. I think so far already done 3. That's about 60% is being done. And validation wise, we can say we've done 50% because I already validate the features microRNA that's being chosen from the features. So the other part will be for the model testing. So on the very next part, I'll do a disease preparation for Viva also. So this is the Gantt chart. Okay. You may want to take a photo if you want to ask later. So that's pretty much ends my presentation today. Yeah, so I'll open for Q&A session.
Okay, thank you, Leo. Okay, maybe we can start with Dr. Nick. Any question or suggestion?
Okay, hi Leo. Thank you, Dr. Sasi. So wow, a very comprehensive presentation, I think. Thank you very much, Leo.
All right. Regarding your data, right? So you actually started with the 3 database, and then you end up with only 1 databases. So you choose only from the GEO database, right?
Yeah, but this is 2 studies.
Yeah, 2 studies. Yes. Okay. So did you really confirm this is actually the Myrna sequencing, microRNA sequencing data, because you show that it stated non-coding RNA, so there are several types of non-coding RNA, right? But you want to focus only microRNA. So have you confirmed, is it really microRNA database data set?
Yes, because I searched with the query name microRNA.
Okay, all right. So as long as it's confirmed, then should be no problem lah. All right, okay. And you trying to differentiate different type of skin cancer, right? I don't remember what type is it. Basal, what type?
Basal cell carcinoma.
Yes. Okay. And now you proceed with the 2 data set.
Yep.
Yeah, okay. So inside the data set, it has all these type of cancer?
One data set for, sorry for I'm not labeling it here, but I can tell you that, why it never go, okay. This GSE 245676 is the study for basal cell carcinoma, and this is the study for melanoma.
Oh, melanoma, only 2 type of cancers?
Yeah, because I did actually manage to find some other study project as well, but that's like I mentioned earlier, because due to the time that they study, the nomenclature is not that not updated, so it ends up that the microRNA is not consistent at all. That's what I found.
The one
like for
that
example,
okay.
This one. Okay, so the updated nomenclature for the microRNA is actually this. This indicates the mature micro RNA, and no one is using these stars anymore. But some of the studies, of course, this is only for the data for TCGA. I actually, for the combined sheet, actually I have all the combined data from across different study samples. They have these things. So this actually is no people use anymore, but there's data for that. So that's the reason. So that's why I try to filter out from there.
The one that you mentioned, there are several types, not 2 types. I think that's several types. Is it in your objective? Basal cancer, is it on your objective? Because you're now getting only 2 type of skin cancer.
Yeah, the objective is actually to explore on different skin cancer data type. Okay, maybe I should mention here. But yeah, the objective is actually to explore different skin cancer type. But during the exploration, the microRNA is not consistent, so that's why I filter out from there. So maybe I would change my objective.
Because my concern is that you just only have 2 type of skin cancer data set.
Yep.
Right. So maybe you can discuss further lah with your supervisor. Okay. Maybe Dr. Fatma want to continue first.
Hi, Leo.
Yo, hi.
Yeah. Overall, congratulations. I think this is the 3rd time listening to your. Can you hear me?
Yeah, of course. Yes.
Okay. So I think this is the 3rd time for me to listen to your presentation. I think this is the best. The best that I could hear from you. I wish these are the style that you have presented in the beginning.
Okay.
It's only now that we can see the flow, the flow of the work, the flow of the idea that you want to achieve in the end. In the first time during the proposal, I assume you are still new on the project, and then I was thinking that okay, let Leo learn all along the way lah. And then in the second progress, I can feel that you kind of stuck in somewhere there, but maybe because the gap is like a month only, right? And now it's been several months. You have done the work, and I can see that you have quite an understanding on the project, what's needed to be explained, and then you have some elaboration of word and statement on the objectives and the methodology, and it sounds more familiar now.
All right, I'm glad to hear that.
Yeah. Okay. Now, I do agree with the Niyu's comment on the you have a bit here and there. I think now you understand why I asked you about how many data sets. I think in the second presentation, I asked you how many data sets have you now identified? And then that question now is being answered today lah. That the data sets, the database are 3 that you explored, and only from Geo Omnibus that you managed to find 2 data sets that fulfill your criteria, isn't it?
Yep.
Yeah. And unfortunately, that two have come back only to 2 subtypes of the skin cancer.
Yep, that's true.
And then I do agree with Dr. Niyus when she comment you need to revise the subtypes here in your objective instead of 3. I don't know whether you want to explore more on the other data sets or not. But I think for your dissertation, I think it's good enough that now you have a wide and understanding on the flow of the work and flow of the idea. If it's okay with the two subtypes to be used to be predict, I think that's fine. Now, my comment is on the data sets. Maybe in your thesis dissertation soon, you need to provide a table on the character of the data sets. How many samples are they coming from? If it's sequencing, what technology has it been used? What year? And then the background of the patient being collected. If it's mentioned somewhere lah in the data set, sometimes it mentioned if it's Caucasian, white, or African American, something like that. You can see in the data sets, right?
Yeah, but I have a comment because that demographic of the patients, right, is only presented on TCGA. While this Geo Omnibus, right, it doesn't provide, because the sample they are using is FFPE samples or cells culture or tissue culture.
Yeah.
So I can label this, but I don't think I can label for this. But of course, I will label some others relevant information.
Yeah. As long as it's relevant to you lah. I'm just giving an example.
Yeah.
Okay. Okay, and then so far this is supposed to be your final presentation.
Yeah.
And then after today, you actually supposed to work on the write-up and whatnot. But from my experience, it's always not like that lah for the mixed smoke student. You still have a lot to do, and then usually the end of the day, it sounds like everybody is stressed. I've done the final presentation, but I have a lot of work to do, and then I still need to catch on the
am I lagging? Oh.
Ming,
I think you're lagging.
We wait for Dr. Fatma.
Yeah, it's lagging. She's lagging.
Okay, yeah. Oh, okay. Dr. Fatma is here. She will continue to.
Okay.
Sorry, Leo.
No problem.
Yeah, my laptop went off, and thankfully, Dr. Nikyu's room is just next door. So, yeah. So yeah, I understand the stress. I mean, you have some more work to do in which you haven't tested your data sets on the model, isn't it? That's what I understand.
I'm on middle way to build my model, but I haven't fully done that objective yet, so that's why I did it present today.
I see, because I assume if you don't present it here, even though you have like five or six model, five?
Five models, five prediction
model. And then you did mention you have tested them on 2 or 3.
Yep.
But I can see that you haven't shown us here lah, meaning that you're not ready to tell the world what's the result of that model, isn't it?
Yeah.
Yeah. Okay. So so that actually the results of the model is actually your final results finding, isn't it?
Yep, that will be, and that will be taken to do validation.
You have one more, is it 1 or 2 months more to go?
1 month plus, I guess.
1 month plus, but at least it doesn't cover wet lab. It's just you with your powerful computer to finish everything, isn't it?
Yep, that's true.
Yeah. Okay. Now, the data validation, when you use the term data validation, usually we test it with a real sample, isn't it?
Oh, okay. Yeah, I think so.
Yes.
Okay.
Yeah, because I think your work, you wouldn't be able to test your model on the real sample, kan?
Yep.
Yeah. So that's another comment that maybe you need to change a bit your objective. I don't know, maybe you should discuss that with your supervisor, because in our case, the term used for data validation, meaning that we test our, we test our essay, we test our key, we test our model, in your case model lah, on a real sample.
Yep.
So maybe need to revise a bit on the objective write-up sikit lah. But I can see now there's a nose there, the 2 eyes there, the face is getting there. Unlike the 2 previous presentation you had, in which it's a bit, you sounded like a bit lost, but now I can see everything is being written down properly, and I can see in the end of the day you will have a thesis lah. Okay, so
how long do you think you can take for the validation process?
Actually, it's very fast. It's just
Very fast.
script
Validation or the model?
Yeah, I just need to study some script, and then to find out how can I do the script, execute the script to do. Yeah.
And you saying that you started with 2000, right? 2000 microRNA.
Yeah, 2000 plus.
And then you filter until
30
1000. 30 something?
Yeah. After
30
doing this
something.
lasso regression, I only get 30 something of microRNA types.
30 something microRNA. That one is belong to what type of cancer? Basal?
Some are from basal cells and some are from melanoma. Do you want to see the combined sheet?
I just want to understand your data. How are you going to differentiate between basal and melanoma cell? Basal and basal.
Oh no. And here, right, this is only choose like which controlling variables I'm going to use on later. But of course, I will also refer back to the original data set and filter out some of the irrelevant one and left with all these all columns that with all these.
Yeah, not many, right? Not many microRNA actually.
Hmm.
Tapi okay lah. I think the other I've heard of AI, AI machine learning, and then they are running validation samples at 12 microRNA. And they have the specificity, sensitivity more than 70-80%. So that's okay lah. So if you have 30, but like I said, validation, you probably need to run on real samples. So the term validation itself, I don't know if with 30 you want to run it on what? Sequencing.
Oh, you mean how do I validate all this?
Validate if you were to validate.
Okay, actually, like I mentioned, okay, let me show you the script lah, how I do this. Actually, I attached in this here. So let me go to the lasso. Okay, over here. So I'll talk about some data preparation, how I do this. This is the script of how I execute the method that I mentioned just now. So by loading some library, I'm using R for your information. I'm using R Studio. So this is the library that contains some of the important function or some what you can say features that I can use to do my things. Okay. So on this line, of course I load my data and then I make sure I do in a format that is readable. Now at this point, I do the number of bootstrap iterations for 1000. So that means that I have run this process, this feature selection process for 1000 times. And the accuracy is being measured from this histogram. So yeah, so you can see that the data actually is quite accurate for the number of times that I run, 200 times that is reached over 95% accuracy. So yeah, did that answer your question?
Okay, I think looking forward for your Viva nanti lah, but Viva we won't be there, right? Viva we won't be there because it will be your examiners. But this is our first time listening to a more detail on the how you run because it wasn't here during the first and second presentation, and now you are sharing it here, so we need some time to digest it or to comment it later. But anyway, this is final presentation already, so I don't think we have that luxury to comment later on. But so far you progress well, and then I hope you the best lah until the end for December or February.
All the best, Leo.
Yeah, no problem. Don't worry, I will show you how I got all these things. I'll keep the script and then I'll run again on the computer during the Viva so you all can see how I did it.
Okay, as long as layman can understand it lah. I mean, at the moment, we, only you can understand it, and probably your supervisor and your co. Okay, back to you, Dr. Sasi, Dr. Neus, you have anything?
Okay, thank you.
I have a question.
Yeah.
Yeah.
Did you count how many times of so you said just now?
Oh, I did.
Not so, the long one. So.
Oh.
Maybe you can get the recording and listen back to the recording. How many times you say the word so? Many times. Okay.
Yeah, sure. No problem, doctor.
Okay, if there is no question, thank you, Leo, for your nice presentation. Wish you best of luck, and thank you all. So with that, I'd like to close today's session. Thank you.
All right. Thank you very much, everyone.